Alrighty. Um, Good morning. Good morning. I believe we can start now. We have a very long way to go. Welcome to the Professional Exam Online Workshop for the year 2025. So this workshop is intended to assist with preparations for the next upcoming exam, right? So we are already in the month of May. We have 3 exams coming up for the professional exam. We have the applied practice in the legal framework on the 26th. Then we have SUTIX on the 28th, and then we have pharmacology on the 30th. We write our exams every second day, Monday, Wednesday to Friday, and the aim here is to really aid in preparing. So we will look at all the 3 papers with specific attention to the second paper, which is the pharmaceutics and pharmaceutical chemistry, because we have noted that that's where the most of the struggle is. And then we will spend the last hour assisting you prepare to enter into the actual exam, since I saw that most of the people that have booked to write at their place of residence. So then I would need to show you how to get into the exam on the day and how you would be invigilated on the day. You are speaking or you are listening to Lisbeth Gumede. I have spoken to most of you via email or WhatsApp, and today I'm hoping that we will be able to finish this workshop. It should take us for about 3 hours. Please be patient. We will have body breaks in between. And please make sure that you stay up until the end, especially because that's when we're going to be looking at how to log into the exam. So firstly, we will look into the content and the format of the examination. We will break down the exit level outcomes, which is what your paper is based on. So your paper is based on the exit level outcomes of the BFARM program. And so we will need to ensure that you sort of kind of understand what those are. Then we'll look into preparing for the practice in the legal framework paper, the pharmaceutics and pharmaceutical chemistry, applied pharmacology, and then the exam platform, how to access your results, what do you do if you're not happy with your results, and then we'll have a Q&A. With the Q&A, we will have another Q&A at the end of the pharmaceutical chemistry and again at the end of SUTIX before having an overall Q&A for everything else. So once the facilitator for pharmaceutical chemistry is done, I will give 5 minutes for the Q&A, and then also when pharmaceutics is done, another 5 minutes, and then we'll do a proper Q&A at the end. Sorry for that. So the professional exam, number one, it is an open book examination, right? So what that means is that you can prepare all your resources, whether they are hard copy or electronic resources, and bring them into the exam. The entire exam is actually 300 questions split over 3 papers over 3 days. All our questions are multiple choice questions, right? And the minimum pass mark for all of them is 50%. We do not apply negative marking, which means that ensure that each and every question is answered before you submit, because you might lose the mark by not answering, which you might gain if you do answer. So on day 1, or on the 26th of May, where we write the practice in a legal framework, you are writing an integrated paper that looks at pharmacy administration, professional pharmacy practice and ethics. You will be having legislation, case studies, and practice scenario questions. That paper has 120 multiple choice questions. All our questions are multiple choice. You have the question with 4 possible options, and then that paper is written over 4 hours. So like I said, it's 2 papers. So you need a sub minimum of 40% in the practice questions, 40% in the ethical questions, and then an overall pass mark of 50%. On day 2, we will write the SUTIX and pharmaceutical chemistry. Also 2 parts. One part is the SUTIX, second part is pharmaceutical chemistry. Over 3 hours, 90 questions. There you need 40% per section and an overall of 50%. On the last day, the 30th of May, you will write pharmacology, also over 3 hours, 90 questions with a pass mark of 50%, right? So like I said, your paper is based on the 11 exit level outcomes of the BFARM program. However, because number 11 is research, we will not be asking you any research related questions within the paper. So we will only be focusing on level level outcome 1 to 10, right? And then you will see that with the actual paper, which is all 3 papers combined, we have split it across all the 10 exit level outcomes. But then when you go into the different sections of the exam, you will notice that different papers focus on different level outcomes or level ELOs, and as we go along we will then breaking it down. When we get to the pharmaceutics and pharmaceutical chemistry, the facilitators will also be helping you focus more on the ones that are in their paper and giving you specific examples per ELO. So when we look at those different exit level outcomes, you will find that the first one is basically around the fundamental science principles and knowledge of pharmaceutical sciences. The second one is knowledge of product development and formulation in compounding, manufacturing, distribution, and dispensing of pharmaceutical products. The third one is looking at how you compound, manipulate, and prepare medicines in accordance to GMP, GPP, and GCP. The fourth one is looking at how you manage manufacturing, packaging, and the registration of pharmaceutical products. The fifth ELO focuses on logistics of selection, procurement, storage, distribution, and disposal of pharmaceutical products. Number 6 is how you dispense medication and ensure optimal pharmaceutical care. 7 is how do you apply a pharmaceutical care management approach to ensure rational medicine use, while number 8 is all about how you initiate and modify therapy where appropriate and making sure that you're keeping with accordance to GPP and GCP where they are applicable. Number 9 is all about the principles of public health. 10 is how do you integrate and apply management principles in the practice of pharmacy. And then, like I said, 11 is participate in research. However, you will not be getting any questions on that. So your focus is only on ELO 1 to 10. When we try to break those ELOs down a bit further, the first ELO, which constitutes 12% of the entire paper. So it's 12% of all the 3 papers, being ethics, pharmaceutics, and pharmacology. With that one, you are focusing on the physical, chemical, and biological principles and how they're integrated and how they're applied in development, formulation, and compounding of manufacturing, drug supply, and dispensing of pharmaceutical products, right? There you must show that you are able to demonstrate ethical and professional conduct relating to the scope of practice of people that are supposed to be providing pharmaceutical technical support services. Then you also focus on the anatomical, physiological, biological, and pathophysiological principles. You also look at social and behavioral principles and knowledge and how they are integrated and applied in the initiation of therapy and provision of pharmaceutical care. With the second ELO, which constitutes 9.67% of the entire paper, your focus is on the physicochemical and biopharmaceutical principles and how they are applied in the formulation, developing of pharmaceutical products. This is where you look at the picking, the packing, and you're making sure that all your orders are secured. You will also focus on the physical, chemical, and biological principles. You look at physiochemical and biopharmaceutical principles and how they are applied in compounding and dispensing. And then pharmaceutical products are also, you ensure that they are produced within integrity and it is maintained within the entire GPP principles. With the third ELO, which is 6% of the paper, there you are focusing on how SOPs are developed from GPP. You will look at pharmaceutical preparations that are compounded in accordance to GPP, sterile mixtures, and how to record your products and actually store those records. Number 4, which has 10.33% of the entire paper, you will look at medicine registration dossiers, master production documentation, all the GMP requirements, how the different dosage forms are manufactured in a laboratory scale according to plans and standards. You will focus on packaging, label, and packaging inserts. You will also look at a quality management systems. Number 5, which is 6.33%. Here you're looking at the selection of medicine and related products and how it is managed according to the rationale, scientific and evidence-based principles and patients' needs. You will look at the quality of medicine, the procurement of medicine, pharmacoeconomic knowledge, the storage and distribution of medicines, and the disposal of expired and unwanted pharmaceutical products. Number 6, 16%, you are looking at how to evaluate a prescription, how to prepare medication, how to provide appropriate drug information, pharmaceutical care plans, record keeping in accordance to GPP. Number 7, which is 14.67%, you are looking at all the philosophies and the principles of pharmaceutical care and how they are demonstrated, the pharmaceutical care management approach, rational drug use, and pharmacovigilance. With number 8, which is 11.67%, you will be focusing on relevant clinical information, all the appropriate advice. In the case of medicine interactions, you will look at contraindications and appropriate modifications, and then also how do you keep medicine records or appropriate records. With number 9, which is 8% of the paper, you are looking at disease prevention and disease management tools that are used to inform the public on healthcare and lifestyle modifications. How do you do promotion, health promotive services? The public health is assistant to recognize and manage healthcare risks, screening tests and how they are also managed. How do you keep records when you are doing disease prevention and disease management? With number 10, which constitutes only 5.33% of the paper, you are looking at basic financial management, human resource management, strategic management principles, marketing management, logistics management, relationships with patients, caregivers, and other healthcare professionals, risk management, and other quality improvement principles and strategies that are continuously applied within the management side of pharmacology. And then when it comes to how our questions are structured, we use different levels, right? So your entire paper will be a mix of straight up knowledge questions that you can find from your resource material or simply remember from your head, or there will be the next step of questions, which is your application questions, wherein you are applying the knowledge. And then the third level is your problem solving questions. Please note that these questions will not be stated that this is a knowledge question or application question or problem solving question. However, it becomes your responsibility to recognize that this is a knowledge question straight up. Are you supposed to be applying certain knowledge to get to the answer, or is this a problem presented to you that you must then solve in order for you to select the correct answer? Again, all of our questions are multiple choice questions. You have one question, 4 possible answers, and you select only one answer. Right, so the aim of this presentation or this workshop is to assist you prepare, right, for the exam. So we've already communicated the dates and times of the exam. We said that it's the 26th, 28th, and the 30th of May, right? And then the second opportunity would either be in October or next year. The venue, when you were booking, you were able to select where you would like to write. A majority chose that they're going to write at the comfort of their homes, and then we will now be assisting you prepare the resources that you will need on each day of the exam, right? So how do you prepare? You attend the workshop, you go through our website, and you look at all the available resources there. What references do you need? Some of it's going to be shared in this presentation today. The rest you can find in the brochure that is available on the website, right? And what you need to bring on the day of the exam, you will need a calculator, you will need 2 devices, which is a laptop and a cell phone, and you might need a piece of paper and a pen to scribble, but then we will also focus a bit more on that as we go. How does the online examination work? We will answer that towards the end. How long is the exam? The first paper is 4 hours, the other 2 papers are 3 hours, right? But then we'll break that down as we go as well. So like I said, it's 3 papers written over 3 days. We will now pay a bit more focus on the first one, which is the applied pharmacy practice in a legal framework, after which we will then look at the pharmaceutics and pharmaceutical chemistry, paying specific attention to each section, and then we'll end it off with the pharmacology. This is based on how they are written on the specific days. Right, so the first paper is 120 questions in 60 questions, which is 50% of the paper, is pharmacy practice. The other 50%, 60 questions, is law and ethics, right? So remember I said that the questions are a mixture of knowledge, application, and problem solving. So with this paper, you have less than 5% of it being straight up knowledge, more than 50% of it being application questions, which are your case study or scenario-based type of questions, and then less than 45% of them being problem solving. When it comes to references that you must be familiar with on the day, right? You need to be aware of all possible legislations. With the legislations, you are able to find most, if not all of it, on our website. So if you go to the South African Pharmacy Council website, right, somewhere on the far right, there's like a book sort of kind of icon. That's where we have all our legislation. You will find the act there, you'll find all the rules, you'll find all the regulations. So what you can do is you can go there and download everything and have it in a specific folder for you to access on the day. You can access them on the day as electronic versions or online versions. However, that might take you a bit more time to do the navigation. So my advice is you go there and you download all of those legislation that we have on our website. Also be familiar with the PSSA Pharmacy Law Compendium, both volumes, right? Look at the South African Medicine Formulary. Make sure that you have the SAMF, the latest edition. Have access to the standard treatment guidelines, right? You can also access them on the website there on the Department of Health. Have access to community pharmacy, the one that focuses on symptoms, diagnostics, and treatment. The 4th edition should work. Have access to a book in pharmacy management, that specific one by Alston or any other. Have a communication skills in pharmacy practice book, and you can also have any book that will be able to provide you with some knowledge on diagnosis and therapy. So you can have the MEC manual, have the daily drug use, and have symptoms in pharmacy. So what we're saying with these references is try access them as soon as you can and go through them and familiarize yourself with how they are written, so that you are not opening them for the first time on the day. Know which information is in which reference material. What you do not want to do is spend so much time trying to figure out which resource will give you which information, right? So if you bookmark your resources, then you will know with this question, I can go to this resource. Also try to prepare as much as possible to be able to answer some of those questions without having to go through any reference material. So if the paper is 4 hours, that is 240 minutes. You have 120 questions, right? With 4 options per question. That means you have approximately 2 minutes per question. This is not enough time for you to reference each and every question. That is why preparation is key, so that you are able to answer some of those questions within seconds based on memory, and then the rest you answer them via checking your references. So time is of essence because we do not extend times. If the 4 hours is complete and you are not done, unfortunately, that's the end of the road. So preparation is key, right? I will now we will now look at the applied pharmaceutics and pharmaceutical chemistry, right? Wherein Prof. Lisa will now take us through the pharmaceutical chemistry first, right? Pharmaceutical chemistry has 40 multiple choice questions. It is spread across ELO 1, ELO 2, and ELO 4. So she will take us through it, and she will also give us examples per ELO, what to focus on in the questions, and how to prepare for that particular paper. Prof. Lisa, over to you.
Good morning, everyone. Thank you so much. Can you hear me?
Yes, Prof, we can hear you loudly.
Thank you so much, Ms. Gumede, for the introduction and for having me here today. I have a bit of a flu, so please do excuse my voice today. So as Ms. Gumede highlighted, I will be going through just a brief overview of what we cover really in the pharmaceutical chemistry paper, and then just give you a few examples. These are very representative examples, so it would be important for you to actually have a look at them and see if you can as well answer them after this. And then provide you with some key reference texts as well. So I'll just share my screen. Okay, so you saw already the breakdown in terms of the split between pharmaceutics and pharmaceutical chemistry. So pharmaceutical chemistry, because it is, it's a basic science that's integrated then within pharmacy, it is very much focused within ELO 1, with some aspects of ELO 2 and ELO 4 as well. For pharmaceutical chemistry, there are 40 questions. The level of cognition is 20% knowledge questions, 40% application, and 60% problem solving. So this should already show you that with pharmaceutical chemistry, there's a lot of focus, especially on calculations and important application questions where it's not just a straight answer from the reference text. Some kind of evaluation is required within the question. So pharmaceutical chemistry, why it's so important for us as pharmacists, practicing pharmacists going forward, is we need to have that knowledge of the drug actions and how that controls drug delivery across biological barriers so that it can reach the receptor site to have its mechanism of action. To do this, we need to develop stable formulations and be able to analyze these formulations and drugs as well. So in order to do this, we need an understanding of basic physical, biophysical, and organic chemistry. So this is important for the development of your small drug molecules and for biotech products as well, which are increasingly coming into the market, your peptides as well as your monoclonal antibodies, for example. The importance of pharmaceutical chemistry is hinged on the fact that we need that study of your inorganic and organic compounds according to their pharmacodynamic grouping, so in accordance with the actual mechanism of action. And really what we focus on a lot is your sedatives, hypnotics, narcotics, NSAIDs, tranquilizers, psychotropic drugs, anesthetics, hormones, vitamins, chemotherapeutics, antibiotics, and radiopharmaceuticals. What's also important in pharmaceutical chemistry is the synthesis of these drug molecules, as well as the chemical reactions that are used for the identification. The biotransformation pathways are also very important, as some drugs may be metabolized to a more active product or a more toxic product. We need to know the structure-activity relationships of these drugs, which is how the actual functional groups on that molecule are related to its biological activity. So why do we do this professional exam in pharmaceutical chemistry? That's so that we can see that you have the knowledge of the basic principles of your organic pharmaceutical chemistry. So to break it down, you need to understand the basic principles of your pharmaceutical chemistry and structure-activity relationships that influence drug actions. So that is the principles of organic chemistry and the structures of organic compounds, as well as the reactions of these functional groups in the synthesis of these compounds, as well as the structural determination of compounds that you do not know, so that you need to identify basically. Very important is the quantitative as well as qualitative chemical analysis of substances of pharmaceutical importance. So, quantitative would be determining, for example, concentrations, pH, etc. Qualitative would be actually identifying functional groups, for example, using FTIR. And then organic, medicinal, and pharmaceutical compounds, we need to place a special emphasis on the correlation of the structural, physicochemical, and chemical properties, obviously with their biological activity. So this is where the connection comes in with your pharmacology. You need to know drug sources as well, mechanisms of drug action, drug design and selectivity, and drug incompatibilities are also very important. So being able to pick up any notable drug interactions. And pharmaceutical chemistry actually shows you the basis of these incompatibilities and interactions by showing how the functional groups actually interact to, for example, form possibly a toxic product or a product that doesn't have any activity, because certain drug molecules, when administered together, can inactivate each other. So general principles in molecular mechanisms of drug action. What you need to have an understanding of is the general principles of drug action and pharmacological activities of various classes of drugs. And where it ties in here now with chemistry is focusing on the molecular mechanisms of drug action. So how functional groups are related to that specific drug action. So a very important component of the pharmaceutical chemistry exam is the analysis of substances of pharmaceutical importance. So this is a lot where those calculations and interpretations are going to come in. So weight and volumetric analysis is very important. So looking at neutralization, precipitation analysis, and complexometry, mass spectrometry, TLC, HPLC, GC, infrared analysis, ultraviolet, nuclear magnetic resonance spectroscopy. So knowing also where these would be specifically applied in the analysis of drug molecules. And then, of course, very important is your calculation and interpretation of properties of chemical solutions, like your acid-base properties, your molarity. Molarity calculations, you'll see a lot of those. The actions of drugs in biochemical terms. So you need to have an understanding and be able to interpret as well the actions of drugs in biochemical terms. So their structures, functions, and transformations within living cells, the structure and function of drug molecules, biopolymers, nucleic acids, and proteins. So these are all therapeutic molecules. The effect of drugs on metabolism and regulation of cellular processes is also important. With specific regard to pharmaceutical chemistry in the design of drugs, molecular orbital theory in drug design is very important. Electronic and symmetry properties of molecules, so your isomeric forms of drug molecules as well, conformation of drug molecules, and the actual impact on drug action. Because we know that if you change the conformation of a drug molecule, for example, if it's transformed to its bioisomer, for example, will it still have an interaction with your target site, with your receptor site, and elicit a drug action? Okay, so that was just some of the background. Now we're going to just go through some actual examples. So this is a very common question type where we're looking at the degree of ionization of a drug molecule when exposed to a specific pH. So this would be very important. For example, now if a patient is taking an aspirin tablet and knowing the degree of ionization, and this would help you to be able to assess as well the permeability of the drug molecule and the solubility of the drug molecule. So, key points that you need to note in this question. So, it would be helpful to just highlight those as you're going through your actual exam. So, you're given the pKa of aspirin. You're given the pH of the acidic media of the stomach. So now you have to basically figure out what is the applicable equation for this. So very important once again, and as Ms. Gomede highlighted earlier, it's important to know where to look for your references and for, not having to like open millions of files to know where something is or go through millions of books. So it's important beforehand to have an idea of the types of equations you will be using as well, so that you can quickly do these calculations rather than having to look up the calculation. So have the calculation readily available to yourself. In this case, because it's an acid-base scenario, and we're looking at the degree of ionization, we're using your Henderson-Hasselbalch equation. So what also becomes important now, and this is where you need to be very capable of using your calculator and how to do various transformations on your calculator. Write down the equation. You know that your aspirin is an acidic drug, so for an acidic drug, pH is equal to the pKa plus the log of your ionized component over your unionized component. And then you substitute in those values that were given to you. Now you need to transform the log so that you can actually calculate your ratio of your ionized to your unionized. So it's then the anti-log of your 3.5 divided by 1.5, and then that gives you 0.01. So that's basically 1 over 100. So that shows you that of the total component, 1% is ionized and 99% is unionized. And you know that the unionized component has permeability across your barriers, specifically in this case, your gastrointestinal barrier. So your unionized component is 99%. Okay. So another very important question is calculating either molarities or molecular weights of drug compounds. So here you need to be very aware of the equations that you would use. I mean, I would already have those available in memory if possible. So here you presented with an unknown amount of the antifungal miconazole. The molecular weight is given, and that was dissolved in 50 milliliters of solvent to give you a solution with a concentration of 0.041 molarity. And you're asked to determine the mass of miconazole to form the solution. Very, very important with your calculations is to always be cognizant of the units and make sure that once you're entering them into that equation, that you're using the same units throughout. Okay, so always remember molarity basically is moles per liter. Okay, you see you're given the solvent amount in milliliters, so that would have to be then transformed into liters to fit within the equation so that you can get your conversions correct. Okay, so we'll know your concentration is equal to the number of moles divided by the volume. Number of moles can then be transformed into the mass over the molecular weight. So that whole equation then transforms into concentration equals the mass over the molecular weight multiplied by the volume. Okay, so the concentration is given to you. Just remember always, like I said, that molarity is moles per liter. You're given the molecular weight, which is in grams per mole, and then you have to transform that 50 milliliters into liters. So that's very important in your equation in order for you to get the correct mass. Okay. Now this shows you more of a qualitative analysis, and a very important question type that often comes up is determining the hybridization of your atoms within a drug molecule. So here they've asked for the hybridization of the nitrogen atom. And you can see in this form in the molecule, it's primary amine. And what is important to note with your amines is the lone pair of electrons. So that is what influences the actual hybridization in this instance. So you have basically 4 attachments, so sp3 hybridization here. Okay, so note that you could also be asked, for example, hybridizations of your carbon atoms as well. So as an exercise, maybe have a look at these 2 and predict the hybridization here as well. Is it sp3? Is it sp2? Okay, so go through your molecular orbital theories for actually having a knowledge of your hybridizations of your specific atoms within drug molecules. Okay. Drug isomers is another very important topic. And as highlighted, certain isomers can have similar activity, they can have a greater activity, or they can have no activity. So here we've given the molecule tetracycline and its isomer epitetracycline. Here you can see that the alpha configuration of your amine is changed to the beta configuration in epitetracycline. Okay, so now what you need to do is then look at the definitions of these terms and see which would apply to these isomers. So diastereoisomers, the definition is compounds that have the same molecular formula and sequence of bonded atoms, or bonded elements, atoms, but they are non-superimposable, non-mirror images. Now, if you look at the definition of what epimerization is, it's a process in stereochemistry in which there is a change in the configuration of only one chiral center. So if we look specifically at tetracycline in acidic conditions around pH 4, tetracycline undergoes epimerization at position 4, and an inactive isomer is formed, which is your 4-epitetracycline. Okay, so here we kind of see we're on the right path there in terms of what these isomers are to each other. Just looking at the other definitions though, regioisomers, these are constitutional isomers that have the same functional groups attached at different positions of the molecular backbone. So you can see that this is not applicable in this case because all the functional groups here are within the same positions. There's just your change here at position 4 of your tetracycline molecule, where you've had epimerization from the alpha configuration at your nitrogen to your beta configuration. Epitetracycline is also not a prodrug of tetracycline. So the answer here would be your diastereomers. Okay, so as I mentioned as well earlier, biotransformations of drug molecules are also very important for knowing if the effects of metabolites as well of your drug molecules and how they are excreted. It's also important as a pharmacist to know, our molecule is the molecular metabolite of the molecule excreted via the kidney or the liver. Of course, in patients that have kidney issues, you'd want to rather prescribe a drug that's more focused on metabolism in the liver. Okay, so first thing here is you need to recognize the molecular structure of the basic molecule. And the question already has given you some guidelines because it's talking about codeine and morphine, and these are within your opioid class. So now what you need to do is figure out, okay, is this codeine or is it morphine? So basically, if you're looking up this in your text, the reference text, if you'd go to the opioids, you'd see that codeine is the N-methylated version of morphine. Okay, so there already you have a bit of a start. Now we have to figure out the positioning and the type of biotransformation that has happened here. Is it so it's already given you a clue that it's glucuronidation. Conjugation as well can also occur, and this is with a sulfate group. So we already know glucuronidation, and at what position is this occurring? Okay, so basically what you need to then do is go to your reference text if you're not already comfortable with actually counting the positionings on your molecules and numbering the molecule. So is this position 3 or is this position 6? Okay. Have a look and figure that out. And then we'll have a look at the answer. Okay, so this is position 6 of your morphine molecule. As I highlighted earlier, there's no methylation on your nitrogen group, so it can't be codeine. And we have attachment at position 6 of your opioid molecule with the glucuronide molecule. So you see this is a very interesting molecule as well. It looks like there's 4 rings, but you can see there's an additional ring that's coming out of the plane. And this nitrogen group is the center of bioactivity of your opioid molecules. So very often you'll be asked to identify a molecule and maybe either identify the molecule or say which pharmacological class it belongs to. So this in terms of its bioactivity. Okay, so having a look initially at this molecule, I'm sure some of you can already see the base molecule or the base component within this molecule. Okay. What is very important is that you already see here your beta-lactam 4-membered ring. So that should give you an idea already that this is a beta-lactam, so it could be then a cephalosporin or a penicillin. What differentiates your penicillins from your cephalosporins? That's the ring that's attached to your beta-lactam molecule. In penicillins, it's a 5-membered thiazolidine ring, whereas in cephalosporins, it's a dihydrothiazine ring. So that was pretty easy. You can see that this belongs to the penicillin class of drugs. Sometimes you can actually then be asked to identify the actual molecule. So you already have some indicators here by the methoxy groups that are attached to the ortho position there in your acyl side chain of your penicillin drug molecule. So this specific structure is actually methicillin. So what would be really important for you here is to actually already be able to identify that this belongs to the beta-lactam classes and narrow it down to penicillin. So then if you are asked what specific molecule it is, you could then go to your reference text and look under the penicillins to be able to identify the structure as methicillin. Okay, you can see your basic structure for your penicillins, your beta-lactam ring attached to your thiazolidine ring with your acyl side chain. So the substitution in the acyl side chain here is what differentiates your molecules. Specifically for your penicillins, you have various attachments at your position 6 acyl side chain. And the ortho position of the methoxy groups, like I said, highlights it as methicillin, which is beta-lactamase resistant because of these methoxy groups that come out here like arms, protecting the molecule from beta-lactam degradation. Okay. Another common question which is asked is how many stereo centers exist within a drug molecule. Okay, so how many atoms with rotatable bonds exists within this molecule? So I'll let you have a look at the structure for a little bit and see if you can count them all. The specific molecule given here is an antiviral, zanamivir. Okay. So let's see how many of you managed to identify the number of stereocenters in this molecule. 1, 2, 3, 4, 5. Okay, so you can see rotatable bonds exist around these 5 atoms within this molecule. So the answer is 5 for zanamivir. Okay, so here we have another calculation type question. And calculating the concentration of a drug molecule based on its absorbance is very important to be able to do for characterization of concentrations of drug molecules within unknown solutions, for example, after you've measured the UV absorbance. UV spectrophotometry is a very common quantitative analytical technique, which is often used by pharmaceutical scientists for actually characterizing and determining the concentration of drug released at specific time points. Okay, so what important information is given within this actual example? We have the molecular weight. We have the molar absorption coefficient. So this is already also giving you an idea of the type of equation that you're going to be used. And the wavelength is provided, but this won't be important for your actual calculation. And then you're given the concentration. So what you need to calculate here is actually the absorbance. So you're kind of working backwards here, because usually what you're calculating would be that concentration. So very important to note is the units here of your concentration. So that will be important when you're inputting it into your equation. Okay, first step, what equation would we need to use here? It would be Beer-Lambert's law, which is the absorbance is equal to the molar absorptivity multiplied by the path length and the concentration. Okay, so what I indicated was that the concentration would need to be transformed so that it fits within your equation units, because as you saw in the previous slide, your concentration, your molar absorption coefficient is provided, which is in liters per mole. So it's the inverse of your molarity. Okay, where the path length is not given, we assume a path length of 1 centimeter. Okay, so you transform your concentration in percentage weight per volume, which is grams per 100 milliliters, to your grams per liter. And then because your molar absorptivity is provided in liters per mole, we have to now transform this once again to your molarity. So you're taking your concentration in grams per liters and dividing that by your molecular weight that was provided to get your concentration in moles per liter, so that it once again fits within your equation units. Okay, and then after you've done your transformations, it's very easy to input that into the equation to get your absorbance of 0.4. And from this calculation, you'd kind of already know that you're within the right space in terms of your absorbance, because generally when you're doing quantitative analysis by UV spectrophotometry, you want, for accuracy, you want your absorbance to generally fall within a range of 0.1 to 0.9 for the best accuracy in your measurements. Okay, here's another example now of qualitative analysis. So important physicochemical characterization technique is your FTIR. So knowing the regions where specific stretches are is important. So if you can find yourself within your reference texts, a nice table where it shows you where specific stretches occur for each functional group, at which wave number, that would be very important. Okay. So you're given a number of options here, and you need to now narrow down the range. So your reference text will highlight to you that carbonyl stretching peaks are strong and they occur between the region of 1800 and 1600. So basically, we know there that we're going to look at a specific range for your carbonyl stretch. Specifically, the carbonyl stretch of your aliphatic esters occurs from 1750 to 1735, whereas those of your unsaturated esters is from 1730 to 1715. So your correct range for your carbonyl stretch would be at 1730. Okay, here's another question where it's asking for the pharmacological classification of the attached structure. And you're given quite diverse options there. Okay, so how do we even start? How do we narrow this down? You need to have a general knowledge of your basic structures of your drug classes. So, before we looked at your antibiotics, so having a basic idea of how cephalosporin looks, how a macrolide looks, and when other classes that are very important are your cardiovascular drugs, your anxiolytic drugs, your antidepressants. So in order to be able to identify the structure, you need to have a general idea at least of the basic essential compound. And here we have the very important identifying feature is you have a 7-membered ring. And even more important than that, you can see in that 7-membered ring, you have 2 nitrogen groups. And when we're naming a structure, nitrogens are generally azo compounds, so diazo. So you have a 7-membered diazepine ring. So that should already now take you on the path as to what class of drug this molecule belongs to. I showed you the 7-membered diazepine ring, so that already leads you to thinking along the lines of benzodiazepines. This specific benzodiazepine is brotizolam, and what would already give you an indication of that is that you have the bromine group attached to the central ring structure, your central benzodiazepine ring structure. Okay, so that just shows your lead compound structure of your benzodiazepine, 7-membered diazepine ring, generally attached to a ring with saturation. Okay, this is a very important question which requires your evaluation skills in coming up with the answer. So This is asking which macrolide lacks the C6 hydroxyl group. And because of this, it doesn't then undergo your internal ketal formation, which is very common with your macrolides. And this ketal formation, the ketal is a toxic product which results in gastrointestinal cramping. So here you'd have to actually have a look at these structures and go to a reference text where it's talking about your macrolides. Have a look, which one does not have the hydroxyl group at the C6 position. Of course, for this, you'd have to then have a good knowledge of your numbering of your drug molecule as well. So just looking at this question now, I don't know if any of you have some idea of which of these macrolides does not undergo internal ketal formation. Okay, so very important, looking at this question, which of these molecules are actually macrolides? Only erythromycin and clarithromycin. So that rules out 50% of the question already. Now to identify which of these, erythromycin or clarithromycin, lacks the C6 hydroxyl group. So erythromycin was one of the first macrolides discovered in the 1950s already. And as you can see, at the C6 position, it has that hydroxyl group present. In clarithromycin, that is converted to a methyl ether or a methoxy group. So because you don't have the free hydroxyl group here at position 6, it's actually not going to undergo that internal ketal formation and will be free from causing any cramping if it's administered just as is. Clarithromycin was actually developed around 1991, so many years after erythromycin. So interestingly, and also an important fact to know, so if you tie this in with your pharmaceutics, for example, how would you then, what pharmaceutical approach would you use to then administer erythromycin so that it doesn't undergo that internal ketal formation on oral administration? So if you just think about it, what you could do is develop a tablet form, dosage form that's enteric coated. So then it wouldn't undergo that internal ketal formation in the gastric environment and be targeted for delivery to the small intestine, where it would have better absorption as well. Okay. So now just to have a look at some of the very important reference text for your pharmaceutical chemistry. Looking specifically at drug design and drug metabolism, we have drug-like properties and the organic chemistry of drug design and drug action. You just need one or the other, you wouldn't need both. You'll need a good textbook on pharmaceutical analysis. For example, fundamentals of analytical chemistry, pharmaceutical analysis, pharmaceutical calculations, introduction to pharmaceutical and chemical analysis. A good organic chemistry textbook is also very important. McMurray's has always been good. When you're looking specifically at structure-activity relationships of drug molecules, your 4S principles of medicinal chemistry is very good, and also all be a good text for looking at biological classifications of your drug molecule. For example, if you're identifying a specific molecule within a drug class. Otherwise, an introduction to medicinal chemistry. Very important for specifically spectrometric identification of organic compounds is the textbook by Roberts. So very important tips when you're actually answering your questions, and we have gone through this as we look specifically at some of your calculations, is to select the correct formula. So basically, like I said, have those important equations like your Henderson-Hasselbalch, your Beer-Lambert's law equation, your molarity calculation equations. Keep those on hand. Always check that when you're doing your calculations, that you've written down the value correctly. And then, of course, when you're actually changing the units, for example, that you've done so correctly, check your decimal places very carefully all the time when you're changing from one unit to another. Arrange for your answer, that's also very important. Okay. Ms. Gumedi?
Thank you very much, Prof.
Thank you so much.
Do we have any questions? I'm not seeing anything on the Q&A. However, you are able to unmute your mics. So if you could maybe raise hands, we identify and we unmute, or we use the Q&A. Prof. Lisa is one of the people that actually sets the pharmaceutical chemistry questions. So if you have anything with that regard, now is a good time to ask.
Ms. Gumedi, of course, as well, if they'd like to email me with any questions, I'm happy with that as well.
Okay, thank you, Prof.
Thanks so much.
All right, if we don't have any questions, our next facilitator is only available at 10:30. So I guess I was hoping that we're going to have questions from now until then. If you do remember anything, please type it in the chat, and then we can start with that when we come back at 10:30. But if nothing is happening, that's also fine. Like she said, you can email them to me, I'll forward them to her, and we can get that sorted before you write the exam. So let's take a 21-minute break or 20 minutes break and be back at exactly 10:30 to begin with pharmaceutics with Dr. Boka. Do get some water, leg stretch, and we will continue at 10:30. Thank you very much, Prof. Lisa.
Thanks so much. Sorry.
Welcome back. Welcome back. I hope that we are all back from our break. I will now hand over to Dr. Boka for the second session. There's a hi on the chat. If there's any question that you'd like to ask, please do so. Otherwise, we will now hand over to Dr. Boka for us to continue with the second session. Welcome, Dr. Boka. Please do take over the screen whenever you're ready for us to do the pharmaceutics. Thank you very much.
Thank you very much, Lisbeth. I hope you can see my screen.
Yes, Prof, we can. If you could just please put on presentation mode.
Okay, yeah, sure. Yeah, sorry for the background noise. I'm actually in another meeting, so I hope it is not really impacting. But nonetheless, thank you very much for all your those who are around here today. I just wanted to quickly check something before I can put it back on the presentation mode. Okay, right. So I'll be going through the pharmaceutics aspect of the professional exams I'm sure you have had so far from your presentation in terms of the various ELOs that you will be covering across the 3 different papers and actually 4 different papers. And then with the pharmaceutics, I would like to emphasize on what are the ELOs that we will be mainly focusing on. If you look at the column that is highlighted, pharmaceutics has got 50 multiple choice questions, which is mainly covered in ELO 1, 2, 3, and 4, and with a lot more emphasis on the ELO 4, which is about manufacturing, the core aspect of the pharmaceutics. So we will be breaking these things down to say that what are the aspects that you should be looking or focusing quite a lot, especially when it comes to each and every ELO. Yeah, as mentioned, there's 50 questions in total. And then if you look at the breakdown of these questions, very few questions are knowledge-based, where they are straightforward questions, but most of the questions are application-based. As you can see, more than 50% of them that are case-based or scenario-based, where concepts are integrated. So it's not like a single concept that you can look into and answer a question. You might be applying your knowledge in order to resolve multiple concepts to get the answer. Then we have problem solving questions, which are about 40%. Most of the problem solving questions, I would say, these are the pharmaceutical calculations. I will show you the list of the calculations that we will be focusing on, and emphasize on few of the calculations that you might not be aware of or you're not looking into. So that will give you a basic idea of what pharmaceutical calculations that you should be looking at. So that is the breakdown of the 50 questions in terms of the difficulty of the questions or the Bloom's aspects of the questions. To start with the ELO 1, which is basically for the 14%, if you look at it, it has got quite a lot to look at it in terms of the physical, chemical, biological principles are integrated. As I said, that these are integrated. It's not like you get a very straightforward questions. Yes, the knowledge questions can be straightforward, but most applications are, you'll be integrating the concepts of physical, chemical, biological aspects in the development of formulation, compounding, manufacturing, drug supply management, and dispensing of the pharmaceutical products. Being the pharmaceutics, we mostly focus on the development, formulation, component manufacturing. Yes, we do also get into the supply chain management depending on the type of the question. I would like to drag your attention to look at the following bullet points, which are quite important, which comes really handy when you are going to write this paper. When you're looking at the one which up there, the yellow can be broad. I'm just trying to narrow it down a little bit to say that just look into integrating the physical pharmacy aspects, such as your unit operations, like mixing, milling, drying, granulation, compression, coating, and all these unit operations you should be looking at, where the physical aspects and are of those unit operations can be integrated in developing the formulation, compounding, and so on. And you should be looking at the rheology, which is the flow of materials. You should be looking at the micrometrics, such as your border flow properties, thermodynamics in terms of the polymorphism, amorphism, the solubility, dissolution, stability. So these are the main critical aspects under the physical pharmacy that you should be focusing on in order to answer the questions within this particular ELO. Some of the main references I've highlighted at the bottom, which is the Martin's Physical Pharmacy. Hence, I'm saying that you should be focusing mostly on the physical pharmacy aspects. And then you have the golden Bible of the pharmaceutic, which is your Alton. So these two does really help a lot because these have the information pertaining to the aspects that I have mentioned about there, such as unit operations, rheology, micrometrics. I'll be taking you through a few questions. You will see in terms of the difficulty of the questions what I mean by integration. Not a straightforward question. If we look at the first question, the question says, the highly soluble amorphous material formed due to quench cooling of the crystalline drug showed a glass transition temperature of 40 degrees. Which of the following would negatively impact the solubility of the amorphous form? So one should be knowing what is the glass transition temperature, what is its effect on the storage, okay? As much as you know the solubility, you should know that the amorphous materials are highly soluble than the crystalline material, but they also have the thermodynamic instability. And hence, the glass transition of the amorphous materials does play a significant role in terms of the stability. So if you look at it, there's quite a mouthful already in terms of the concepts to understand that what is an amorphous material looks like. I mean, like, what does it mean? What does it impact on the solubility? And then its stability. So knowing that, so from this, if it has a glass transition of 40 degrees, means if you look at your storage, that's what it's trying to say, that which of the following would negatively impact the stability of the amorphous form. So a glass transition temperature refers to temperature at which it changes from into a high stability form, which is like into its crystalline format. So storing at below 40 degrees would not really negatively impact because it's not reaching to its glass transition temperature. Storing at 25, also the same case, it's not reaching to the glass transition temperature in order for it to move from one form to the other form. Storing it as an amorphous solid dispersion, you should know what is a solid dispersion refers to. It is basically to improve the stability of any amorphous form, or mixing with the polyvinyl pyrrolidine, which is also a polymer that is used to make the solid dispersion. But I think if you look at the answer, storing at 40 degrees, which is exactly at its glass transition temperature, that is where it now starts moving from one phase to the other phase, from amorphous phase to a more stable crystalline phase. So I'm just giving an example for you to understand that it's just not a single concept, it's multiple concepts. One needs to apply thoroughly the knowledge that you have in order to answer the question. So we'll take one more question. Second question, which is again a scenario-based question. If you look at the scenario, in the case of water-soluble dyes added to granules prepared with wet granulation technique, which of the following drying equipment would best suit to prevent migration of the color? So there again, multiple aspects there. There's a water-soluble dye, there's granulation aspect, and then again the migration. So one should know what is the impact of drying these granules. And when you have a water-soluble dye, how it moves within the granule from one point to the other point during the drying process. If you use a traditional tray dryer, the conduction of the heat from bottom to top, as it goes, it starts making the water-soluble dye to travel from one point to the other point and concentrate at the tip of the granule. And once you have at the tip of the granule, you see color variation within your granules. So that is the knowledge that you should be applying to say that what can give a uniform heat supply to these granules to prevent the migration of the color. One of the answers, the best suitable answer in this case will be the fluidized bed dryer. As you fluidize the granules, the heat is applied across the granule equally so that there isn't much of the migration of the color because this is a water-soluble dye. In case of water-insoluble dye, you don't have any issues of that one because it does not move along with the water as it is evaporating. So that's not an issue. But the issue here is that you're using a water-soluble dye and it has a tendency to migrate along with the water vapor and then come to the tip of the granules. So I cannot be able to give you all the questions, but I'm just trying to give you a gist to say that in terms of your preparation to the exam, you just don't expect a very straightforward question. You rather look at integrating multiple concepts. So you need to understand the concepts within the pharmaceutics and the physical pharmacy, the terms, that the definitions, what does it mean? Then you can apply that knowledge in order to resolve the questions. Moving on to ELO2, which is slightly bigger, plus in terms of the weighting, which is 24%. So here we basically referring to the biopharmaceutical principles, which is your pharmacokinetics, pharmacodynamics, the bioavailability aspects. Again, the solubility dissolution can come into picture in this aspect. So if you further look down on into this particular ELO, we'll be focusing on the biopharmaceutical factors, as I mentioned, that affects the formulation design. For example, like patient factors, drug-related factors, all these things. How do they play a role in terms of the formulation design? And the pharmacokinetic aspects, as I mentioned. If you have a clinical situation, how do you apply the pharmacokinetics of a given drug in terms of the volume of distribution, clearance, area under curve, and all these things. Then we'll also be focusing a little bit on the novel drug delivery systems, focusing on the gastrointestinal drug delivery systems, colon drug delivery, nasal drug deliveries, including the vesicular drug delivery system. What I mean by vesicular drug delivery systems is your nanotechnology-based drug delivery systems, such as your liposomes, niasomes, things like that. Those are the vesicular drug delivery system. Apologies for the mistake there. They say drug is instead of the drug. So the focus, as I mentioned, we will be looking at the biopharmaceutical factors impacting the formulation design. We'll be looking at the pharmacokinetic aspects that should be taken into consideration to resolve clinical situations, such as determining the dosage regimen, the amount of the dose based upon the half-life, the clearance, and so on, volume of distribution. And then again, novel drug delivery systems to say that how they can impact the therapeutic outcomes based upon the different types of the novel drug delivery systems. The references are laid down there. Your focus is mostly on the Shergill, Applied Biopharmaceutical Pharmacokinetics by Leon Shergill, and also the Drug Delivery and Targeting by Perry. So those are the books that you can look at it to have an understanding of these concepts. Again, just taking through few questions of the scenario based. Which are the following age-related factors can significantly affect pharmacokinetics among elderly patients that would lead to dose adjustments? I think this is a very straightforward question. There is nothing much complicated in this. Not many of the concepts are involved. The age-related factor that can significantly impact increase in total body water, decrease in body fat, decrease in serum albumin concentration, decrease in creatinine clearance. I think that's very straightforward answer in terms of the biopharmaceutical questions that we're referring to. Maybe moving to a little bit more complicated questions. As you can see, multiple concepts here. There's a pKa, there's a pH, there's a log piece that can be used in order to resolve, in order to understanding the drug regimens. So the below figure shows the plasma concentration terminal profiles of the weekly acidic drug X, which has got a pK of 4.0, administered orally as a tablet under fasting, pH 1.2. It shows in red, that is the plasma concentration curve, and fed condition at pH 4, which is in blue. Which of the following statement is correct pertaining to the absorption of the drug? So in order to resolve this, what you should be knowing is that you should be knowing the uses of pH-pK concept together. It comes with the Henderson-Hasselbalch equation. So understanding the pH-pK concepts, if you include them into the Henderson-Hasselbalch equation, you should be knowing that what is the ionization aspect of this drug at various pHs. And knowing that unionized drug is more absorbed compared to the ionized drug, so one should be able to find out whether it is more ionized at pH 4 or more ionized at pH 1.2 or less ionized in that way. So that's what actually lead to give you an answer to say which of the following statement is correct pertaining to the absorption of the drug. The rate of absorption is always faster in the fasting condition compared to fed conditions. The drug taken under fasting conditions result in 100% ionization, leading to greater Cmax. How do you know that? By applying the pH and pK into a Henderson-Hasselbalch equation and then you get to say whether it is 100% ionized or 100% unionized. So that is how you will be getting to the answers. So questions can vary. Again, as I say, it's not that this is exactly what you're going to get, but you will be integrating the concept. That's what I'm trying to refer to time and again in my presentation. So I think that's where the problem also with the many applicants that are sitting for this exam, expecting it a very straightforward questions in pharmaceutics, like which of the following is true or which of the following is false, kind of. No, it's more of integrating multiple concepts into reaching to the answer. One of the calculations, the problem-solving questions, as I mentioned, most of the problem-solving, around 40%. I think that's what you should be also looking at it in terms of scoring. Around 40% of the questions are problem-solving, which are calculations-based. So don't leave any stone unturned or any kind of the calculations you didn't work it out before you come to the professional exam. I'll put up the list again at the very end. I have the list so that you can also go through the list to familiarize yourself, to work on those calculations questions. But just in this case of the biopharmaceutical aspects, one of the questions that I can just show you is that calculating the bioavailabilities. I'm just giving an example. Or maybe you given a half-life, you calculate the volume of distribution, you calculate the clearance rate. So all these calculations, I'm sure you have done during your B.Pharm as part of the pharmacokinetics and pharmacodynamics, which is your biopharmaceutics modules or the semesters. So if the bioavailability of the sildenafil citrate is 50 milligram tablet is 0.25 compared to the bioavailability of 0.75 in a sildenafil citrate simple syrup. So if you look at it, there are two different dosage forms, the tablet and the syrup. And I'm sure one should be knowing that the bioavailability should be high for a syrup compared to a tablet because there's a dissolution, disintegration, all those things comes into picture. If you're substituting, this is quite commonly happens in terms of substitution when you don't have a tablet in the hospital, then you will be substituting with a syrup. Then how do you calculate the bioavailability factor? How do you use the bioavailability factors in order to calculate the dose of the syrup? So those are the things that you should be looking at the formulas to say that how do you calculate the absolute bioavailability, relative bioavailability. And if you have the bioavailability factors, how do you calculate the dose substitutions? So, yeah. So if you look at the amount of sildenafil citrate bioavailable from the tablet is so on so 50 milligram tablet multiplied by its bioavailability factor, which is 0.25. Means if you take one tablet, you get 12.5 milligrams of sildenafil citrate. Whereas when it comes to the syrup, it says 0.75, and it has got a dose of 1 milligram per mil. So each mil, you'll get 0.75 milligrams. It's a very simple question. So you need to get 12.5 milligrams. And hence you can calculate how many mils you will be required. So that is a cross multiplication. And you get 16.7 mils of the syrup is required in order to be equal, to provide the same dose that you get from a tablet. Moving to the next ELO, which is ELO 3. This is where we basically focusing on the pharmaceutical preparations are component in accordance to GMP. So the focus is mainly on the GMP, good manufacturing practice, good clinical practice, not good laboratory practice, or GPP, good pharmacy practice. But the focus for the pharmaceutics is basically on the GMP. When I refer to the GMP, one of the standard references I would like to bring to your attention is the GMP guidelines, which is the essay guide to GMP. You can get this thing on the SAHPRA website. SAHPRA is the regulatory authority of South Africa. And if you go onto the website, you should be able to get this GMP essay guide to GMP book, which speaks about the 5 Ps, which is like your premises, personnel, procedures, processes, and documentation. The 5th P is actually the documentation. So it does speak in terms of the guidelines pertaining to that. Then one can focus on the validation and qualification, installation, the qualifications like your IQ, OQ, PQ, the validation, which is your process validation, cleaning validation, analytical method validation. But you really don't have to go very deep into that. But I think I'm trying to understand what are the requirements of a process validation. For example, when we refer to process validation, one of the main requirements is that one needs to conduct process validation on 3 consecutive batches in order to establish the critical attributing manufacturing factors. So you should be trying to understand what is the validation from a process point of view, from an installation point of view, or from an equipment point of view. Then one should be focusing on the cross-contamination. How do you minimize the cross-contamination? For example, if I can say, in terms of the cross-contamination, one cannot manufacture cytotoxins and the normal drugs in the same facility because they can be highly cross-contaminated, or penicillins in that matter. You cannot be manufacturing the penicillins and the normal drugs in the same facility because of the cross-contamination. So one should be looking at how do you minimize cross-contamination. You can also focus on the requirement of the audits. What is a regulatory audit? What is an internal audit? What is a quality audit? What is a third-party audit? So you should be looking at different types of audits. And then also product recalls from a South African point of view, to say that what are the guidelines of recalling a product within South African context. So you can also get these guidelines on the SAHPRA website. So you should also be focusing on that. The second aspect under this ELO to look at is the sterilization techniques. So we'll be focusing on the sterile products, formulation, the sterile pharmaceutical products such as your small volume parenterals, large volume parenterals. Again, this is a pharmaceutics aspect completely. Okay, as much as we have the GMP aspect, we have also combined the parent sterilization aspects into this particular ELO. So look at your dosage forms that are pertaining to sterile dosage forms. That can include ophthalmics, that can include parenterals, both small volumes, the large volume parenterals, nasal formulations, and most important thing that you need to look at is the isotonicity adjustments. How do you calculate the required sodium chloride or required isotonicity adjusting agent? Because we do have some calculations pertaining to that in this particular under this particular ELO. I'll show those calculations as we go. So you can see the references also laid down for this particular ELO there, but emphasis I mentioned is that looking to the SI guide to GMP, the GMP guidelines pertaining to cross-contamination, validation, and qualification. Taking you through few questions of the GMP. According to good manufacturing practice, sliding doors may be undesirable in clean room because they are difficult to open and close under pressure. They are very expensive to maintain. They may have uncleanable recesses, cannot handle pressure differentials. So one of the suitable answer is basically from a GMP point of view, cleaning point of view, is that they are difficult to clean because they can have uncleanable recesses or areas where you cannot approach to clean them. And also if you look at the question, it says that undesirable in clean room. Clean room refers to something where it's extremely clean in terms of probably manufacturing sterile products in that room. So one needs to have, I'm sure you might have learned this in your B.Pharm or taught you in your B.Pharm to say that if you're manufacturing any sterile, the premises of the sterile should be in a way that they should be absolutely accessible to all parts to clean easily. One more question. Good manufacturing practice requires that materials and products should be stored accordingly to avoid contamination, mix-ups, and instability. Which of the following statements we're trying to move away from is or are? There will be only one statement. Which of the following statement is true pertaining to storage conditions used? So basically, you're trying to avoid the contamination and the mix-up. So if you look at the options, intermediates of effervescent product should be stored below 25 degrees and 50% relative humidity. I don't know if you do know the requirements for storing the effervescence. Effervescence should be stored below 40% relative humidity. Rejected material should be stored in lockable cage. This again comes from the guidelines of the GMP. Starting materials can be stored with intermediate products as long as the temperature and humidity conditions are maintained. I don't think that is correct, because you should be separating both the starting materials, intermediates, and the finished products, and they stored separately to prevent the cross-contamination and mix-ups. Finished products can be stored in a passage as long as they are shrink-wrapped to avoid mix-ups. You cannot. This is also part of the guidelines as you read through the GMP, that you cannot store them anywhere because there is exposure to the sun and it has got an impact on the stability of the product. So looking at the different options, the rejected material should be stored in a lockable cage. That is the true. Okay. Another question, which of the following parameters need to be optimized during the process validation? Remember, I spoke to you about the process validation of a tablet, dosage form manufactured by wet granulation. So when you are doing the process validation, what is the purpose of process validation? Is to optimize the critical processing parameters. Okay. So the question says, which of the following parameters needs to be optimized, whether it is quantity of the diluent, storage conditions of the finished product, container and closure system, loss on drying. So one of the aspects is that the loss on drying that you need to be, these are the critical parameter. Like when you add the granulating agent, then end of the day, you checking the loss on drying to say whether it is meeting the required loss on drying or not. The amount of moisture that should be left inside the tablet. This is one of the questions that from the sterile products, parental products that we refer to, which are the following excipient is not advised to use in the formulation of large volume parental products, where it's a cyclodextrin, benzethonium chloride, dextrose, sodium acetate. I think the answer, as you see, that benzethonium chloride is because of its toxicity for a long-term use, and especially when referring to the large volume parental products, you cannot be using the benzethonium chloride, which is a preservative. Okay. Moving to the ELO4, you have multiple aspects within ELO4. The first aspect I would like to start is the medicine registration aspect with SAPRA. So one should be looking at what are the CTD requirements, which is common technical document requirements of both the API and the finished product. You should be looking at the guidelines on stability to indicate that when do you store, how many batches do you have to store for the stability, how much data you need to supply to SAPRA in order to release the product. If it is a generic, how many batches? If it's a new chemical entity, how many batches? What are the batch sizes? What are the different conditions of storage that one needs to put through it? So you need to be focusing on the stability guidelines. You should also be looking at the dissolution guidelines to say that when do you perform dissolution? Do you perform dissolution for the BCS class 1 or BCS class 4 in terms of the biowaiver? So look at the dissolution guidelines also, and post-registration amendments. So once you have registered a product, and if you wanted to change any aspect of the product, for example, let's say you wanted to change an excipient in the product, then how do you submit a post-registration amendment to SAPRA? What are the different types of post-registration amendments are out there? What is its impact on the quality and safety efficacy of the medicine? And lastly, the other important aspect to look at it is under the medicine registration is the impurity profiles. What are the different impurities one can come across in the product, such as organic impurities, inorganic impurities, heavy metals, residual solvents? What are the guidelines stating about in terms of the impurity profile? For example, like nitrosamines. Nowadays, we're also looking at the nitrosamines as very critically in terms of the impurity profiles. So one should be focusing on the CTD. How many modules are there? Module 1, 2, 3, 4, 5. What is the importance of the modules in terms of the information that is going inside there? As I mentioned, the focus can also be on the stability guidelines, dissolution guidelines, guidelines pertaining to post-registration amendments, and the impurity profiling. What are the impurity profile requirements as per the guidelines? These are the quite critical aspects. There are many, but I'm highlighting very, very important and critical aspects that you should be looking at as part of this. So if I can just give an example from a stability question, how many pilot scale batches should be considered for stability testing of codeine tablets? So when you look at this question, we're asking to say that how many pilot scale batches? One should know what is a pilot scale batch or a production scale batch. You should be knowing the definition of it, which is part of the guidelines again. Should be considered for stability testing of codeine tablets. The question says codeine tablets. It refers to a product that is already existing, means it is a generic product. Which, by giving the name, we're saying that it's a codeine tablets, it's a generic product. So you can go onto the guidelines to say that how many batches do I need to put into the stability if it is a generic product, basically. So I'm not necessarily directly saying it's a generic product, but we are giving a hint in the question to say that this is a generic product. It's not a new chemical entity. So then the guidelines are straightforward, very clear. It indicates that it needs 2 pilot scale batches in order to conduct the stability studies of a generic product. So similarly, if it's a new chemical entity, we will indicate new chemical entity, definitely, because it's not registered. and that will be informed. And then you can look at to say that how many batches is required. It might be more than 2, it might be 3, depending on the guidelines. So if you look at this question, which is another scenario type questions, PTA Pharmaceuticals decided to use fluidized bed dryer as the tray dryer method was taking too long to complete the manufacturing of paracetamol tablets. So you should be knowing that paracetamol is already a well-known chemical entity. So we're talking about a generic product already. Which of the following minimum data is required for submission to SAHPRA for the approval of post-registration changes? So it's a generic product. It has been registered already, but now you're just making a change on the process of, instead of using the tray dryer, using the fluidized bed dryer. So one need to know that what is the impact of this change in the quality, safety, and efficacy. So you are changing an equipment. The fluidized bed dryer is much faster, and compared to the tray dryer, so it will obviously have an impact on the quality of the product. So what is it that you need to inform SAHPRA? you should apply your mandate to say that what is it that you need to minimum data that is required is a moisture content, obviously, because if let's say that tray dryer you're doing 10 hours of drying, but fluidized bed dryer if you did 4 hours of to finish drying, you should be knowing that the end point is basically the moisture content. Whether you're going to do 4 hours in the fluidized bed dryer or 3 hours with the fluidized bed dryer, the time is determined by the moisture content. So one of the information is the moisture content. And then what is its impact? How do you determine its impact? Whether it is going to have an impact on the dissolution or whether it is also going to have an impact on the dissolution and stability. So one should be looking at to say that it might have an impact because you change the drying method from a slow drying to a rapid drying, which can have an impact on the API degradation. So one of the best suitable also is the type A dissolution, stability, and you need to conduct the process validation because you change the entire thing from a tray dryer to fluidized bed dryer. So those are the aspects that will be informed when as you read through the process validation, as you read through the post-registration amendments. Another scenario type, PTA Pharmaceuticals decided to substitute an active ingredient, an active pharmaceutical ingredient, which is API, of a multi-active tablet dosage form by a different salt form of API. So now you are removing one API. Let's say that this product has got 4 different APIs, and you're substituting one API with a different salt form of an API, where the efficacy, safety characteristics are not significantly different. Which of the following variation type allocation should be submitted to regulatory authorities prior to marketing? So if you're removing an API, substituting it completely with a different salt form of API, it has got a greater impact on the safety, efficacy, and quality of the product. That you should be able to figure it out very straightforward. So one should be looking at to say that you're replacing an API means it's a completely new applications. You're putting a new API of a different salt form. Instead of, let's say, amlodipine, you now add amlodipine bacillate or amlodipine bacillate, which is a salt form which increases the solubility of it, basically. So when you have a different solubility factor altogether, it has got a different therapeutic efficacy altogether. So hence, it has got a greater impact on the quality, safety, and efficacy. So hence, you have to submit as an extension applications. This information is in the guidelines. As you read through it, it says what is the type 1A variation. It speaks to say it has got minimal to no impact. Type 1B, which has got minimal to moderate impact on the quality, safety, efficacy. So that's when you need to apply your mind as well to say that if I make a change, this is what you do in real time to say that if I'm going to change an API with a different salt form, then you need to apply your knowledge to the pharmaceutics to say that how does it impacts the quality, safety, and efficacy. Obviously, it has got a higher solubility compared to the normal drug, which is not a salt form. And hence, one needs to be looking at data like stability data, clinical efficacy data, even the process validation data and all those things. So there's quite a lot of data that is required as you change the API. Hence, it is an extension application. So hence, I'm emphasizing on the guidelines for the post-registration amendments. Then in terms of the same ELO, we have other aspect, which is the documentation. So you should be focusing on the importance of various documents that are used in the manufacturing company, which is like your SOPs. What are the purpose of SOPs? The contents of SOPs. What is the COA, Certificate of Analysis? What does it have? What information does it have? Specifications. What is the importance of having specifications at the manufacturing sites? One should be looking at what is the importance of site master file. What is the site master file constitutes of? What is the purpose of site master file? What is the validation master plan? What is the purpose of validation master plan? To conduct validation, to conduct revalidation, to conduct cleaning validation. So they have all the information going into validation master plans. Look at various documents, their purposes, and how they are used at the pharmaceutical industry for operations. So you need to look at the logbooks. You need to be looking at what are the contents of batch manufacturing and packaging documents. These documents are available online. Various examples are out there. But again, this information as part of the SEC to GMP guidelines is also emphasizing on the need for the SOPs and COAs and things like that. But the contents of it is not in the guidelines, but one should be looking online to say that how does a COA looks like? What information does it have? What is the purpose of having a COA? So just to give a question, which of the following would not form part of site master file for a specific manufacturing site? So if you know what is a site master file, what is the purpose of site master file, the contents of site master file, then you should be able to answer this question. For example, like organization chart, is it not part of SMF? Reports of the finished product analysis, details of starting material suppliers, qualifications, experience, and responsibilities of key personnel. Amongst this, if you look at it, the reports of finished product analysis is something that you generate on a day-to-day basis. A site master file is like a Bible for the site, indicating what products they have, who is working, what are the qualification requirements, and so on and so forth. So hence, if you look at these documents, the importance of these, the contents of this, you should be able to answer this question to say that the reports of finished product analysis is something that is generated on a day-to-day basis. They cannot go onto a master file. A master file should have guidance documents for your day-to-day operations, but not information of day-to-day operations. Like filed into that. Which statement is true of the following regarding documentation in pharmaceutical manufacturing facility? Batch records give directions for performing certain operations. Example, cleaning can be completed after manufacturing process is completed. Packaging instructions should include details of in-process controls with instructions for sampling and acceptance limits. Logbooks are not needed when products are manufactured using dedicated equipment. So again, if you look at the different documents in this question, it's speaking about the batch records. It's speaking about the packaging instruction. It's speaking about the logbooks. One, if you look at these documents and see what are the contents of it, what is the purpose of this thing, you should be able to answer to say that because we are looking at the true answer. Packaging instructions should include details of in-process controls, what you should perform during the packaging process. It's not that batch records do not give directions on cleaning. They give directions on the manufacturing operation, and then cannot be completed after manufacturing process. It has to be completed immediately as you perform manufacturing operations. Logbooks are not needed when products are manufactured using dedicated equipment. Every equipment, it should be having its logbook, irrespective whether it is a dedicated or non-dedicated equipment. So this information, you should be able to grasp it as you read through about these documents. Lastly, I'm sure this is mostly your favorite part, and most of the time in BPharm, you do work on this thing. Your focus is mainly lies on this one in terms of the formulation design, formulation development, where we're focusing on all the different types of the dosage forms like your tablets, capsules, syrups, suspensions, emulsions, suppositories, all the non-sterile dosage form I'm referring to, and also the not the novel dosage form. These are the non-novel dosage forms, which is your traditional dosage forms. As I mentioned, the tablets, capsules, syrups, suspensions, emulsions, creams, ointments, suppositories, aerosols. So all the dosage forms here we focus in terms of the formulation to say that what excipients do they go? What is the purpose of each and every excipient? in the formulation? How do they make an impact on the dissolution, or how do they make impact on the release of the drug? So there's quite a lot, honestly, on this one. But again, one of the most important references that I would like to mention is the Alton. It speaks in detail when it comes to all these dosage forms. So I'm sure you probably will love this section. Which of the following is an advantage of glycerol-gelatin base when used in the formulation of suppositories? So we're speaking about an excipient called glycerol-gelatin base already, as you can see from the example. So what is, when it's used in the formulation, what is the disadvantage? So produce a laxative effect, becomes brittle when cooled rapidly, produce different polymorphic forms when cooled rapidly, has got poor water absorbing ability. So one of the answer I would share that it's the glycerol-gelatin base. When you use it, it produces the laxative effect. That's its disadvantage. So very straightforward questions. So most of the questions are straight, but again here we do integrate to say that if you change this excipient to other excipients, what is its impact? If you include this particular excipient, what is its impact? Or in order to get this particular outcome, which of the following excipients should be used? So something along the lines you can expect questions. So again, if you look at this question, capping of the tablet can be eliminated by, so which is one of the processing problems of the tablets. How do you eliminate it? By pre-compression. One can perform pre-compression, or increasing the final compression rate, means use speed up the machine, use concave punches. You should know what is the purpose of concave punches. What is the disadvantage of using a concave punch? It further leads to the capping. Increasing the moisture content of granules through addition of water. So what happens if you add more moisture? It sticks to the punch and it does not resolve the capping issue. So basically, one of the suitable answer is using the pre-compression, means perform a small compression stage before the main compression. Durability of a tablet to combined effects of shock and abrasion is evaluated by using, is a definition basically. So when you attribute to shock and absorb abrasion is basically your friability. That's what you're checking. So it's a very simple and straightforward question. Yeah, my apologies. So those are the different types of questions you can get under formulation. Again, as I mentioned, that focus is mostly on the excipients. Their purpose, each excipient has got its own purpose at different concentrations. For example, like if you take starch, starch has got different purposes at different concentration. At a low concentration, it can be binder. At a high concentration, it can be binder and a filler. When it is in a liquid form, it has got different binding properties compared to when it is in a dry form. When you use a normal disintegrant, which is your starch, compared to a super disintegrant like sodium starch glycolate, it has got impact on the tablet disintegration and further dissolution. So one need to be looking at different excipients when you get into the formulation. Look at excipients that are used for different formulations. What is their purpose? What concentrations are they used, and why they used at such concentrations? For example, like your the colorants, or when you, not necessarily the colorants, but let's say the preservatives. Some of the preservatives you prefer to use at a very low concentration because you don't want them to impart their quality properties to the product. For example, like if it has got a color, you don't want to use a preservative at a higher concentration, then the product color has completely changed, or it changes the pH of the product. So there's a lot about excipients that one needs to look at it, understand their properties and at different concentrations, and how do they impact the product output, product quality and performance. Coming to the calculations, as I mentioned, this is the major chunk again in the paper, 40% of it. You can really focus on this aspect again. I'm listing out majority of the calculations that are here that you can be anticipating in the paper. For example, like dilutions, you can look at all different types of the dilution-based calculations with like C1V1, C2V2. Or the allegation method is something that I would like to highlight to say that do not leave it behind, because you might be given 3 concentration and you need to make a final concentration. So how do you come up a final concentration given the 3 different concentrations of 1% zinc cream, 2% zinc cream, 3% zinc cream? mix the three of them to make a 2.5% zinc cream. So alligation method comes into handy. So that's something that you can look at it. You should be looking at the concentrations. Again, as I said, C1V1 to C2V2. One of the things that I would like to highlight with the calculations is that in terms of the units, make sure that when you run the calculations, you have similar units. Sometimes we give one in grams, the other one in milligrams or micrograms. So you end up calculating grams versus micrograms instead of converting these grams into micrograms or milligrams, both of them, and then do the calculation. So that is one of the mistakes that commonly done, not converting the units into equal on all sides when you're performing the calculations. So make sure that you focus on that. Then we will be looking at upscaling, downscaling of a master formula. If you're given a master formula, if you need to manufacture now, let's say 100,000 tablets, how do you upscale the batch? That's those simple calculations. We'll be looking at the yield calculations to say, if you start with so much quantity and you should be end up at so much quantity. So calculating the yield, whether you receive 99% or 100% or 98%. So how do you calculate the yields? Then the potency calculations are quite important. As I mentioned, the salt factor. So instead of amlodipine, now you're using the amlodipine bacillate, which is a salt factor. So how do you put that into picture to make sure that the potency is equal? Let's say, for example, you take a tablet of amlodipine versus a tablet of amlodipine bacillate. How much of the amlodipine bacillate has to be added that is equivalent to 5 milligrams of the amlodipine, for example? So those potency calculations to look at. The density-based calculation, the specific gravity-based calculations, to put density and specific gravity into picture to calculate the mass, volume, and stuff like that. Then we have when it comes to suppositories, you should be looking at the displacement values. So if you add a drug which has got specific density, how much of the base has been displaced and how to calculate the amount of the base required, amount of the base required in order to prepare maybe 10 suppositories or 50 suppositories or 100 suppositories. The molecular weights, again, as I mentioned, when it comes to the potency calculations, one can look at to say that if you are using a normal drug versus a salt form of it, how does the molecular weight does impact in terms of calculating the required dosage. I'm highlighting a few things there. Parental solutions, which is isotonicity calculations. So there are two methods basically: sodium chloride equivalent method and freezing point depression method. So these can also be part of the calculations as part of the problem solving questions to say, given a parental solution, how do you adjust the isotonicity? Let's say you're given the sodium chloride or you're given the boric acid. How do you use these? How much you need to weigh in order to make the isotonicity? So those calculations need to be looking at as well. And the other important calculation that I would like to highlight is the shelf life calculations. I'm sure most of you do ignore these things in terms of zero order, first order, second order reactions. So if you have a zero order reaction, what kind of the formula do you have to apply to calculate the shelf life? If you have a first order, what equation you have to apply to calculate the shelf life? And then you should be knowing the last one, which is the HLB calculation, which is the hydrophilic lipophilic balance calculations as part of the emulsions manufacturing or the compounding of it. So these are the highly important pharmaceutical calculations. I think they cover almost everything, but I just trying to emphasize on the isotonicity and shelf life because most of you don't really give attention to these calculations. So don't leave them behind. You can also practice them. So when they're in the exam, you should be able to answer these questions. In that line, I just would like to quickly show you some of the calculations for the isotonicity and the shelf life that you probably can get or was getting in the previous years. In terms of calculating how many grams of sodium chloride should be added to 50 ml of 0.5% weight per volume solution of lignocaine hydrochloride to make a solution isoosmotic with blood serum. So if you look at it, you've been asked to use sodium chloride to make it isotonic of so much amount, 50 ml of 0.5%. Freezing point depression means it's already indicating that you have to use the freezing point depression concept in order to calculate the amount of sodium chloride required. The freezing point depression of sodium chloride and lignocaine hydrochloride at a concentration of 0.5% weight per volume are 0.288 and 0.0625. respectively. I'm not necessarily going to go through the calculations because you have to do the practice of this. If you have not done it, it won't be easy to follow me at this point in time, but there are two concepts, which is the freezing point depression concept, as well as the sodium chloride equivalent concept. So if you can go and practice those two, you should be able to answer these questions pretty easily. Okay, so just to give you, this is a freezing point depression method. As the question indicates, the freezing point depression is given. So you apply the formula of the freezing point depression, which is W equal to 0.52 minus A over B. You should know what is A and B as you practice, and it's a very simple, straightforward substitution. But the catch, often the catch is that the total amount of the volume, which it says for 50 ml. So you probably end up here to say that 0.794% of sodium chloride is required, but that is out of 100 ml. Okay, but you have given 50 ml to calculate. So one needs to complete those calculations to say that, oh, I need to calculate a 50 ml. How much is for the 50 ml? Okay, so sometimes you might not be given 0.5%. I mean, the concentrational solution as 0.5% straightforward. You will be given to say that it's 5 grams in 100 ml, or 5 grams of the lignocaine is there. So you need to convert everything into the required unit. That's what I was referring to, say that make sure that you convert into the required units before you can do the calculation or during the calculation process. That should be kept in mind because the questions cannot be very straightforward. So these things will be changed, and one need to be very cognizant of to say that what units I should be really looking at it before I finalize the answer. Then just another example on the shelf life, just for your attention. The following data represents the decomposition of a drug XYZ. If the rate of reaction is independent of the concentration, look at the tricky part of it here. It's not saying what rate of reaction, is not saying zero order, first order, second order. It's giving a definition to say that the rate of reaction is independent of the concentration, which should say that you are looking at a zero order reaction. That is the definition of a zero order reaction. Determine the rate constant at which the drug is decomposing. So you have the time and you have the concentration at which it is degrading, and it is independent of the concentration, means you need to apply the equation, which is the zero order reaction. If you look at the shelf life calculations, it is part of the Alton also. You should be able to pick up which equation to go with it. And then you just substitute your information into that equation in order to get the rate constant, which is the K. So it might say that next time it might is following first order reaction simply. Then you need to go and check the first order reaction, which is the ln concentration, which is ln C equals to ln C 0 minus KT. So one should understand how do you convert ln C into log C and calculate the C at the end of the day, which is the concentration. So hence it is important that you go and do the practice of using your calculators, convert using the log books or using the calculator to calculate the log values. Okay, so those are the important questions that I could highlight. And just in a nutshell, these are the major references that I would like to bring to your attention, which is the Martin's Physical Pharmacy for all the unit operations. And then you have Alton for all the design and manufacturing of the medicines, especially the ELO4 of the conventional dosage forms. Then you can look at for the calculations, you can look at Ansel, pharmaceutical calculations by Ansel. It has got almost all the types of calculations that I have mentioned. Then you can look at the pharmaceutical practice by Winfield and AJ. It speaks to also the compounding of various dosage forms, like your suppositories, the displacement values, and everything. You can find it there. When it comes to the biopharmaceutics and pharmacokinetics, Liang Shergill is the reference book I would like to suggest, which is also suggested by the Pharmacy Council. Then we have the microbiology aspects, which is the pharmaceutical microbiology aspects by Denier. And lastly, the guidelines, the GMP guidelines, the P&A guidelines, stability guidelines, bioavailability guidelines, dissolution guidelines. All of these things are available on the SAHPRA website. You can also look at them. Then I think the last one, I can't read it here, but that is for the pharmaceutical drug delivery and targeting, specifically for the novel drug delivery systems by Per Revive. So it speaks of all the different types of novel drug deliveries, like your gastroretentive drug deliveries, colon drug delivery, nasal drug delivery, and all those aspects. Yeah, those are the reference books. I can sum it up. I think that is the end of my presentation. And yeah, let me stop it there and hear if you have any questions.
Thank you very much, Dr. Poka. Do we have any questions for pharmaceutics? Anything at all? You can unmute or type in the chat. All right, Dr. Poka, I don't think we have any questions. If they do come through, I know you are in a visit. I will either text them to you or email them to you, and then we can revert back if you need to excuse yourself for the meeting.
All right, no, thank you very much. I wish you all the best to those who are going to write the exam. But as I just wanted to highlight is that don't go with it very much template, but rather have an open mind to say that you will be able to answer any question that can come across to you. And it is an open book system, so you should be able to find your way through answering. But make sure that you go with the mindset that it's not going to be a very straightforward question, and it is going to be integrating concepts in order to apply the knowledge. Again, another suggestion is to focus on the calculations, as it consists major part as well. So practice these calculations as much as possible, so that you can score more there as well. On that note, yeah, that's it from me. I'll wait to hear if there's any questions later on.
Thank you. Thank you very much, Dr. Poka. All right, in the absence of any urgent questions, we will continue then to the applied pharmacology and toxicology. Please do type your questions on the chat. We will get to them. If Dr. Poka leaves, we will email them to him and give us his response.
Thank you very
Prof.
much.
Lisa, thank you very much. Prof. Lisa is still in the workshop with us, and we can also email to her as well. If they both leave, we will make sure that you get your response as soon as possible. All right, moving on to pharmacology and toxicology. So with that paper, it is 90 questions, all multiple choice, and the focus there is mostly ELO 1, ELO 6, 7, 8, and 9. The level of cognition there is less than 10% is knowledge. More than 20% is application, and a lot of it is problem solving, and the paper is 3 hours. The reference material that you can familiarize yourself before the paper is the Basic and Clinical Pharmacology by Katsung. You can have any pharmacology textbook. They prefer the one by Rang HP. Another one that you can use is the one by Harvey. And then you can also have a Goodman and Gilman if you are able to have access to it from any pharmacy near you, or you can buy one, or if you still have one from back in your study days. A pharmacotherapy textbook like the one is a pathophysiological approach, the latest edition by Talbot. You can also have SAMF, the South African Medicine Formulary, the latest edition. You need to also have South African clinical guidelines for various conditions. These you can also find on the Department of Health or our website. And then also make sure that you have our essential medicine list and all the other treatment guidelines, including the pediatric hospital one, the hospital level, and the primary health care level in 2024. Most of these guidelines, you can get them from the Department of Health. Make sure that you download them prior the exam and not during the exam. With regards to all of these papers, right, but particularly pharmacology, make sure that you read the question carefully and you identify the statement which you are sure of and what the question is really asking you, right? Confirm which reference material you need to use and then go to it. But if you know the answer off by heart, try use that, and then you can just go to the reference just as a confirmation, but try avoid using a reference for each and every question, as time might not be on your side. Carry on. if you are unable to answer that question, and know that you'll be able to get back to it towards the end. So if you leave a question blank, that question will be available to you towards the end of the exam. But if you answer it, it becomes automatically saved and you will not be able to answer it at the end, right? So when the question states which of the following is the most appropriate or least appropriate, that means that it implies to the one that is more, that means that more than one of the answers could potentially look correct, but which one of them is the most inclusive answer? So make sure that you look out for those type of things within the question statement itself, right? So with pharmacology, you have 180 minutes, 90 questions, again, approximately 2 minutes per answer. Right. Now we get to preparing for all of the exams. If you go to the SAPC website, right, and this is a page that you should probably be familiar with. It's the same page that you go to for most of the applications. But if you go to the far right end, where you are now looking at the foreign qualified examinations, you will see there that we have a few past papers for you to practice on. The aim of these papers, like this workshop, is to show you the type of questions that you should get, which means that you then familiarize yourself with the concepts around the question, not that you need to now cram or memorize the questions or I don't know, just think that those are the only questions. They are there as examples of the types of concepts and the way in which the questions will be asked. So you should use them to understand the concepts and not necessarily identify the specific types of questions that you're going to be asked, right? On the same page, you will find the brochure for FQ. Everything exam related will be in there. You also have the guidelines. The guidelines are also going to give you a further breakdown on the ELOs and everything that you need to know. You also have the study guide for the exam. So I suggest that you go through all of 3 of these documents. Last year's presentation workshop is still available there, and alongside the presentation, we will also clean up today's presentation and make it available there as well. So maybe you can download the 2 presentations and just go through them with all the material that we try to give you to make sure that you are ready for the day of the exam, right? So make sure that you have the latest reference material, right? You are up to date with current events because these papers are set on the year that the exam is written. The paper that you're going to be writing now in May was set this year in Feb. So we try to make sure that whatever information is within the question is new and relevant, right? So make sure that whatever the new developments are within pharmacy, you are familiar with them, right? Do not use a reference material that you have never used before. Make sure it's a reference material that you're familiar with and you know how to use it. Access SOPs. Some of you have access to the scope of a PT or a pharmacy assistant basic or post basic. So that means that some of you have been low coming. Try and access your SOPs from where you've been getting your exposure from, because you might need to be familiar with certain processes. Make sure that as you are booking, a lot of you have already booked as bookings did close yesterday. A lot of you have booked for the exam. Make sure that on the system, we have your correct cell phone number, your correct email address, and that the profile picture on your profile has been updated in the last 6 months. So please go back, log in, and verify your cell phone number, your email, and upload a picture, head and shoulder picture from the last 6 months. Remember that we communicate with you via email and cell phone also during the exam. So we need to be able to speak to you on the day of the exam as well. If anything changes prior to the exam, if we need to address something during the exam platform, may we please have a functioning cell phone number and email address. Your picture is used for authentication, right? So if you use a full body picture, it becomes difficult to zoom in on your face. Please upload for us a head and shoulder picture. The exam is remote online, right? it means that even if you had booked to come and write at the SAPC offices, you would still be writing it on a laptop online. The invigilation is completely online. So please make sure that, sorry for that, you have a functioning laptop with webcam capabilities, a functioning cell phone with a working front camera, and a QR code scanner. So you already booked for the exam. There is no need for us to go through the booking platform. However, I am showing you this because this is the very same place that you will go back to when now you need to be viewing your examination results. So you see the pathway that you took to get to the professional exam booking. Next to it will be your professional exam results when the results are now released for you to see how you have done. When you were booking here, you had to select a venue. Your venue, your options were home, remote, SAPC or other. A lot of you chose to be writing remotely, which is also fine. You just need to make sure that you have the correct devices for the day and that the place that you chose will provide you with enough quiet space, with enough working space for you to write your exam efficiently on the day of the exam, right? So all you need to do is that while you're preparing, the place must be quiet, right? You must not have noise distractions because noise distractions count as a violation during the exam. You must have enough working space for you to have enough of your material, your reference material. If you're going to be having hard copies, you can also keep your copies electronic if you want to. You must have enough space for you to have your calculator for the Sutux calculations, enough working paper for you to scribble whatever you need to scribble, and you need to make sure that you are calm throughout the paper. Yes, it is a timed exam. However, panicking will not make time move any fast. So you have to try, I don't know, maybe practice breathing exercises or whatever you need to do to remain calm during the entire process. Let's get to the devices on the day. Number one, you need a laptop or a desktop that has a camera linked to it or a functioning camera. We need to be able to see you from the laptop's point of view, right? That means we're seeing your face the entire time. You need to have enough data, minimum 3 gig for one paper. So for each and every day that you're writing, if you've booked for all 3, make sure that on the day that you're writing, you have enough data, 3 gig minimum, have backup data as well. So if you're using Wi-Fi, have backup data on your cell phone or specific router, just in case you get network interruptions. You also need to have a cell phone. This cell phone must be, unfortunately, a smartphone because it needs to have a functioning front camera and it must be able to scan a QR code. Whether it's scanning it via the camera or an app, doesn't matter, but it needs to be able to process a code from a QR code scanner, right? On the laptop itself, doesn't matter what the model of the laptop is, but make sure that it has all 3 browsers, being Google Chrome, Firefox, Microsoft Edge. If there's anything that you do after today is ensuring that your laptop has all 3 browsers, Google Chrome, Firefox, and Microsoft Edge. These are the only 3 browsers with proven compatibility to the exam platform. Others work, sometimes they don't. So we can only guarantee you full compatibility with Chrome, Firefox, and Edge. And we also recommend that you have all 3 of them in case you need to switch in between them during the exam. Right. So the first step is you go to the website, you upload a recent picture, head and shoulder, color photo. This photo is for identification purposes. We need to know that the person writing the exam is the person who booked for the exam. On the day of the exam, we beg that you start the login process from 8. Right? The exam will start at 9 o'clock on the dot, and it will end on the first day at 1, on the second day at 12:30, third day at 12:30. But then we need you to log in between 8 and 9 so that you are able to complete the declaration, you're able to read the examination rule and your rules, and you're able to settle down. So whatever technical issues that you might possibly experience on the day, we have enough time to resolve them. In case there are amendments that need to be done on your devices, they can be done before that, because at 9 the exam will start. And if you are resolving any technical issues, then time is no longer on your side. Right. How do you log into the exam on the day of the exam? So you will go to the SAPC website, go to quick links, then click foreign qualified persons, then click professional exam, then you click on the link here to access the professional exam. Right. We will go through this. If we have time at the end, we can go to the website. But essentially, you just open any browser, being Chrome, Firefox, or Edge. You type the SAPC website, right? And then you go to at the bottom where there's quick links, and then you will click foreign qualified persons. Then you click on professional exams. It will say here, please click here for the professional exams. It will have the 3 different dates on the side. Then you click here. After clicking them, it will take you to this page where you are supposed to now put in your PIN number, your ID or passport, and your password. If you do not have your password, which you should because you were able to book online, I made sure that I do not accept any paper-based applications, because I wanted everyone to know their PIN, their FQ number, their national ID or passport, and their password. So the very same FQ number that you used when you were booking is going to be the same FQ number, whether you used your ID or your passport and your password. They are the very same credentials. If you don't know these credentials, please make sure that you write them down or you request them. But for those that had claimed to have forgotten them, I did make sure that we send you your FQ number, your passport or password, your passport or ID, and your password. So those are the very same credentials that you're going to need on the day of the exam. After having put in those 3 things, you will then be requested to wait for an OTP. The timer will say it will count down from 10 minutes. However, you should receive your OTP within 2 minutes, right? When you receive your OTP, you will then input it there. It should be a number between 4 and 7 digits that you input here, and then you press continue. This OTP again will be sent via SMS to your cell phone number or your email address. So make sure that whatever number we have, you are able to receive SMSs. Whatever email we have, you will be able to access on the day for you to receive the OTP. After having put in the OTP, the system will start doing system checks or equipment checks, right? With the equipment check, what will then be happening is it will check your browser. Are you using any of the 3 browsers? Chrome, Edge, or Firefox, right? So these, all of them, they need to turn green if you are using the correct thing. So if you're using Edge, Firefox, or Chrome, it will then turn green. It will then verify if your laptop or desktop has a camera, right? It will do a webcam check. It will do a microphone check. You need to enable us to hear you, right? So not only must you use the correct browser, we must be able to see you. So if you do not enable your webcam, it will not turn green, it will turn red. If it turns red, there's nothing that you can do. You will not be able to continue. Then it does a microphone check. You need to enable us to hear your background. If you do not do a microphone if you don't allow us to hear you, you will not be able to proceed. It will then check your network. Is your network stable and fast enough for the exam? After that, you do a screen check. With the screen check, you need to share your entire screen, because even though you are allowed to go to a different tab or a different screen, all in all, when you're accessing your electronic references, we need to be able to see what you're doing on your laptop. So you need to be able to share entire screen. If you share screen 1, if you're using a second monitor, it will not allow you to go through, which is why you cannot have a second monitor. You can only use 1 screen, 1 monitor for the exam, the very same monitor that you are sharing for everyone. Then you need to do a last check to make sure that everything is aligned and you are ready to write the exam. Right. There's this link here on the site, or you can even just Google Proctor Edu and do a system check. This is to check your compatibility of your laptop. It will give you whether there's a green here or a red here or anything that you need to fix. I highly recommend that you do this system check, even if it's today, next week, but do this at your own time to see whether the device that you currently have is going to be able to take you to the exam on the day. So please go to Google and Google Proctor Edu, or write down this link and make sure that you test your device's compatibility to the exam platform. So if everything is okay and the system and your laptop is compatible to the platform, everything should be green. Once everything is green here, you will have the next button. If any of these are red, you will not have access to the next button, which means you will not be able to write the exam. Hence the importance of doing the compatibility test in case you need to switch devices. After everything has been done, it will allow you to then verify your name and your surname, your surname and your name. This makes sure that it is exactly what we have on our platform. If from the last time you applied for the for the evaluation till now, something changed, your name has changed, you got married or whatever the reason may be, please make sure that you submit to us an application for change of personal details, so whatever appears here is exactly what should be appearing. Then you'll be able to click the next button. After that, it will ask you to take a selfie. With this selfie, again, the importance of making sure that the picture that we currently have is the same as the picture we're going to get here. When taking this picture, your environment, right, must be of proper lighting. So when you're choosing your venue as well for the day, not only must it be quiet, it must also have sufficient lighting for you to take a picture that reflects your facial features well. Here you might have to tilt your laptop screen a little bit, move around a little bit, get close enough to the camera, but not too close. You must make sure that you are showing your head and shoulders so that we are able to identify you. This is for record keeping purposes, and it's also for authentication. So the place that you're writing at, proper lighting, and quiet enough for you to take this picture. Once you click take picture, it will ask you if you're happy with this picture or you want to retry. You can retry, or you can press next, and it will allow you to move on to the next platform. Once you have taken the picture and you have saved it, you are happy, the system is happy with the picture, a QR code will appear on your screen. The aim of the QR code, right, is to now connect your cell phone. So for the exam, we are using 3 methods of invigilation. The first one is the camera on your laptop for us to see your face while you're writing. The second one is the fact that you're sharing your entire screen, so we are able to see what you are viewing on the actual screen when you're moving to other tabs or other windows. And then the third one is we need to see your entire workspace via your front camera from your cell phone. So how you connect your cell phone is by scanning this QR code, and then on your cell phone, it will ask you to record a 1 minute 360 rotation video of your environment. So you will click record on your cell phone, do a 360 turn, right, of your environment. So you're showing me from your laptop, and then you move around all the way back into your laptop, providing a video of your environment. So I see where exactly you wrote the exam. After that, you will click save. Once you've clicked save, only will you be able to proceed on your laptop. So during the recording phase and everything else, your laptop will be grayed out. There will not be any next button until you record a video with your cell phone of the entire surrounding, and then submitting that video to the exam platform by clicking save. Once you've clicked save, then you will have a next button on your laptop. Once you click that next button, you will get to the declaration. With the declaration, you must read all of these terms, and then you must tick them on the side, stating that you agree before you click on continue. The continue button will not appear until all of these declarations have been ticked. So when you tick until the bottom, only then will the continue button appear, right? So what are you declaring? You are declaring that you are completing this exam in a suitable area with minimum anticipated distractions. That means that you are saying that where you are, you have enough lighting that we're able to see you the entire time and that there will not be any noise. You also declare that the area that you are writing at is the same area that you booked at. So if you booked that you're going to be writing at home, you are going to be writing your exam at home on that day, right? You're also declaring that you have acquired the minimum data required, meaning that you have 3 gigs already for the paper and you will not be saying that you ran out of data during the exam. You also declare that you will not be sitting in close proximity to any candidate while completing this exam. So there's no one who's going to be saying, let's go right together at my place. You will not receive any form of assistance from any person. That can be a pharmacist you know, an assistant you know. In the exam, you will be writing the exam by yourself. You will not communicate, whether it is via WhatsApp, Teams, or whatever, to any other person registered during the exam. You will only use reference materials permitted for the exam, which is the reference materials that we've already gone through for all the 3 different papers. You will not be accessing any prohibited websites during the exam. You will not give any assistance to any other person during the exam. So not only are you receiving assistance, you will not also give any assistance, right? You will not share or retain any content of the exam. So we are able to see when you do screen grabs of the platform during the exam. So you will not be doing any screen grabs and you will not be sharing any content of the exams with anyone, and that you will inform counsel should you notice that anyone or any other candidate contravenes any of these examination code of conduct. After that, you should be able to write the exam. Should it be that you are writing the exam and you are not in South Africa on the day of the exam, please make sure that you adjust your date and time to that of South Africa. So on your settings, make sure that the region is South Africa, the time zone is Harare, Pretoria, and that you have turned off the set the time zone automatically. Because if you are on a different time zone, on a different region, you will not be able to access the exam. Remember, the exam is timed, right? So the time zones must be the same for you to be able to access the exam. Otherwise, it will tell you that you need to adjust your time and date. So make sure before the date of the exam that you have amended your region to South Africa, the time zone is Harare, Pretoria, and that you have turned off the time zone automatically setting, right? When you have finished writing the exam, you will do another declaration wherein you confirm that you have written the examination without any assistance and that you have adhered to all the examination code of conduct, and that you understand that should you be found in contravening any of these conducts, we are allowed to implement disciplinary actions against you, right? And that you will not share anything or retain anything from the examination via whether electronically printed, written, or verbal with any other person, right? Now, when you have gone through the first step, right, and you have logged in and now you are ready to start the exam, please stay on this page until it is 9 o'clock on the dot. The exam will not start before 9. The exam will only start exactly at 9 o'clock on the dot. So you will not be waiting on this page, reading your instructions, and ensure that you are on this page before 9, because you need to be able to start the exam exactly at 9 to be able to utilize your time wisely. If for any reason whatsoever you click start the exam before 9 o'clock, it will tell you that the exam has not started and it will ask you to wait, right? Then you will click OK and wait. Should you be distracted and it hits 9 o'clock and you are not there, At a few seconds after 9, it will tell you that the exam has started. Please start writing. Therefore, you will click OK, thank you, and then start the exam. The exam format is 1 question per page, right? All questions are randomized, so you're not going to be able to see multiple questions at the time. So it's 1 question with these 4 options at the time. The questions are randomized, meaning that even if 2 people are sitting in the same room, they will not be able to get the same question at the same time, right? And it's a timed exam, which is why it's important to make sure that you are on the same region and time zone as South Africa on the day of the exam. Once a question has been answered, you cannot revisit it. However, if you did not answer a question, you will see that question towards the end of the exam, right? So how it looks is exactly like this. The number of the question, how the question will be, your 4 options, right? Here where it says references, we have a few references that we have loaded onto the platform. It's the same references that are available on the website, like your legislation and things like that, that you can access on the day of the exam. But again, we do not really recommend this because we advise that you actually have your references prepared ahead of time. If you click next question before choosing an option here, this question will appear again. If you choose an option and then you click next, this question answer is saved. That means you will not be able to have access to it ever again. Here on the other end, there is a chat with the invigilator. So on the day of the exam, each person is assigned a different invigilator to assist them on the day. This is where you can now speak to whoever is invigilating you on the day of the exam if you have any question. They will be able to assist you with technical questions, but they will not be able to assist you with questions relating to the actual exam question. So you cannot ask them to clarify anything with regards to the question, but they will be able to assist you with login issues, network issues, which reference can you use, what can you do, what can't you do, but they will not be able to assist you with the breaking down or the understanding of the actual exam question. Right. While you are writing, we are monitoring all violations, right? But the nice thing is the violations are visible to both you and the invigilator. So while you are writing, you are also able to see which violations you are conducting, right? So remember I said the place that you are writing at must be quiet, because if there is noise or a conversation, it will tell you that there is a conversation or background noise. And I can also see this violation then, which means that if maybe there's a conversation within the house, you are advised to close the door. If someone is cutting the grass, please close the windows, but try and minimize the noise. If you see a violation, also try chat to the invigilator first and explain to them what the noise is before they reach out to you to also save time. If you are using electronic references on your device that you have shared, it will tell me that your focus has shifted to a different window or a different tab. I will then try and zoom in, or your invigilator will try and zoom in and see what you are doing there. Are you accessing your references from a saved folder? Are you going to the SAPRA website? Are you going to the SAPC website? Or are you going to Google to Google your answer? Then we will also have a conversation with you. So as far as Google is concerned, you can use it to link you or to go to a specific website or a specific reference, but you cannot type a question on Google for it to suggest references, right? So if you are looking for information on measles vaccines, for example, you can use Google to go to the standard treatment guidelines, but you cannot use Google to type measles vaccine for Google to then suggest a reference source, or you can't use Google the measles vaccine for it to give you the schedules or anything like that. So you can only use Google as a search engine for you to get to a reference material that you have already listed there, but not for it to suggest reference materials to you. If you then decide that because you're not comfortable with us seeing your entire work environment and you disconnect your mobile phone or you lose connectivity, it will tell you that your mobile phone is not connected. which you would then need to reconnect immediately before we call you to ask you to reconnect it. If you then decide to connect a second screen, it will also tell you that there's a second screen connected, which we will also ask you to please disconnect, right? If you decide that you don't want us to hear what's happening and you mute your environment, it will also say that you are muted and we'll ask you to then unmute. My advice here, when you see the violation, correct it immediately. It saves time versus an invigilator reaching out to you and then having a conversation to try and fix that, because we will not be able to reallocate that time to you, right? So where can you get help? Via the chat function that I just showed you, that you can please use that chat function and someone will be able to answer you, right? With this function, it's easier because you're speaking to your allocated invigilator, which will have more time allocated to you. You can also send a message to the WhatsApp number. The only issue with the WhatsApp number is that all of you would then be sending the WhatsApp to that one person, and it might take a bit of time for that person to respond. So I know some of you have my personal number, which means that you would be wanting to text me directly. That can work. However, you would need to be conscious of the fact that if everyone is texting me and only me directly on my WhatsApp, I might take a bit of time to get to everyone. If you use the chat function, then you know that you have your own specific person assigned to you for the day to assist you. What if the invitation is to contact you? They will send you a text, right? Or they will email you, or they will call you. All of which we need to be making sure that we have the correct email address and contact number for us to be able to contact you. Or we will send you a message also on the chat function, which will pop up during the examination. If you decide not to respond to us via the chat function, also we are able to see when the message pops up on your screen, and when you close it, we are then able to remove you from the exam. So if you conduct a violation, we speak to you regarding the violation, and you decide to ignore us, we are able to then remove you from the examination while you resolve whatever the matter is. And remember, we will not then reimburse you for that time. So what happens if the invigilator notifies you that you are not connected or your cell phone is not connected? At the bottom of your screen when you are writing, you will be able to see a picture of yourself from at the bottom there, which is the picture from your webcam. You will click on it. Firstly will be the calculator. Second is the QR code, the chat function. This calculator, you can also use it on the day for calculation. However, it is not a scientific calculator, just a basic calculator. So if you want a scientific one, you have to bring one in yourself. If you click on this QR code, you will then be able, to reconnect your cell phone for the invigilation, right? So if for whatever reason there's electricity outage on the day of the exam, please keep calm. Make sure that you then switch to your alternative plan, which I'm asking you to have right now, right? Make sure that your laptop is fully charged before the day of the exam, that you have an alternative connection in case your Wi-Fi does things. And then if you are experiencing load shedding or load reduction, make sure that you have other means, whether it's a second location that you can get to quickly, or it's a second device that is also fully charged, or you have a specific, cell phone that has specific data that you can then use to hotspot on the day, but have contingency plans in place. However, when you answer a question, that answer is auto-saved and auto-submitted. So say you were writing the exam and you had answered 50 questions before you lost connectivity and there was nothing that you can do. Those 50 questions that you have answered will be marked. They are already submitted on your behalf. But then we just recommend that you try getting back into the exam so that you are able to finish your exams, right? So like I've been saying, every answer that you answer, every question that you answer, that question is auto-saved and auto-submitted, right? That is why you cannot get back to it. Therefore, if you get cut, right, due to connectivity or electricity, whatever you have already answered or whatever you already submitted is saved, and you will not be able to get to it, and it will be marked. It's just that you might have to be concerned about what you might not have had time to get to, right? Should it be that you get disconnected, and when you try to get back in, it says that this page has been opened, or if you clicked start the exam a couple of times before 9 o'clock, then what you would need to do is you would need to refresh the platform by closing all browsers and logging in again. Closing all browsers is not the same as closing the exam tab. So when I say close all browsers, I mean close everything to a point where what you see is just your desktop. Then you will refresh it and start the process of logging in again. But the biggest issue that we see is when we say close all browsers, people close the current opened tab. And then when we are busy having the same conversation over and over again, it's eating on your time. So whatever the instructions are from your invigilator, try to follow them as promptly as you can and as directly as you can, because the longer you want to give in different scenarios and opinions, the longer it's going to be eating on your time, right? If for whatever reason you get to this error that says that live proctoring is not connected, close absolutely everything. Refresh your screen 3 times, right? And then start the process again. If you do not do that, this message will keep popping up and your time on the exam will be chowed, right? Again, if maybe you need a refresher on how to clear the cache and the history, should you need to troubleshoot during the exam, please familiarize yourself with the clearing of the history for all the 3 browsers, right? These tabs will be made available. So this is a step-by-step approach to clearing the history on Chrome and how to go incognito in case you need to make sure that the previous history is not recognized. Again, the same step-by-step for Firefox and the step-by-step for Microsoft Edge. Like I said, have all 3 of these browsers available to you on the date, right? So where you sit is very important. Insufficient lighting, right? We can see you. Dress appropriately. Yes, you are writing from home, but we are able to see you throughout the exam. Those records are part of your permanent records. Please make sure that we are not texting you to ask you to please dress appropriately or please sit appropriately, because again, that will be taking from your time. So make sure that the images that we are seeing on the day are images that you can be proud of. We're not saying, be interview ready, but please be decent on the day, right? We've already gone through how you'll be able to see your results on the day. So you will log in like you would be if you're going to book, but then you're going to be in the next door tab where you'll now be seeing your examination results, right? So your examination results are not released immediately because of a very stringent verification process that they go through, right? They have to go through a couple of, they must go through the moderation and then committee before council before they can be released to make sure that everything is correct. So this can take plus minus 2 months. So please bear with us and note that your exam results might take around about 2 months, depending on whether there are any questions that need to be removed. So what then happens is after writing, the questions are moderated. We see who answered what question, what way, what could possibly have been the reason for them to get that answer incorrect. And if there are more people that got that answer incorrect but chose the very same option, then we must discuss again to relook at the question. So all of those things to make sure that the paper was fair take a bit of time, which is also why we do not remark. So we do not remark because in that 2 months, a lot of quality assurance measures are put through to a point where we are sure that remarking will not give you a different mark. What we can do, however, is provide you an opportunity to review the paper. So with the review, you book an appointment. It cannot be done online, and you come into the office. When you come into the office, one of the persons that are responsible for the paper will then sit down with you, wherein you will now go through each and every question with its possible options and the memo and the reference used to get to that. And then you can query, but I use this reference and I found that, and then you can take it forth from there. However, we do not remark, we can only review your paper. Right. If you want to review or appeal your process, please make sure that you submit to council within 30 days of the results being released. Like I said, not a remark, but rather a review. Do we have any questions? I'm sure we all have this email address where we have been sending our queries with regards to the application, the booking and everything. It's the same email address, foreignqualified@sapc.za.org, that you can also, as you are preparing, send through your questions, and then I will make sure that they reach the facilitators for them to be able to assist you with anything that you need. Can I please quickly go with you to the website so that we are able to see where we get all that information I was talking about right now regarding the, so I prefer Chrome. We will then go to Chrome. We can go to the SAPC website. I just want to show you. Here is where you can then get all the legislation that we were referring to. So you have the act, you have regulations, you have rules, board notices, proposed board notices that you can download now, right? And then if you go to the quick links, right? So you were home and then you go to the quick links here at the bottom. If you come to foreign qualified, you will then come to professional exam. This is where you will click on the date to access the exam. Please don't come into the login button, but come here to be able to access it. All the other resources I was referring to, you will then be able to find them here. The past papers for you to get the concept, the brochure, the guidelines, study guide is all here, last year's workshop and presentation. And we will also then be putting the new presentation here when we then get to that. So please make sure that you have reviewed all of this. Whatever other websites you might need on the day, like the SAPRA, you bookmark it for easy access, right? Do we have any question regarding the paper? Okay. There is a question. Can SAPs provide us with all the SAPRA guidelines, especially with the GMP? We're struggling to download them, or even if we can get the link to SAPRA, please. Okay, I will look into the SAPRA guidelines, download and say and send them via email. Either I will send a link via email or I will send the actual guidelines to you. The next question is, are we allowed to bring notes, hard copy we have made during our studying period? Yes, you are allowed to bring absolutely anything that you think will be useful to you. So if you have handwritten notes, you're allowed to bring them onto the day of the exam. Are there any other questions while I try access the SAPRA guidelines? You can unmute and ask your question. I'm just trying to make sure that I send the link to the guidelines now while everyone is here. Oh, I see someone else already put it in there. So yes, we have the guidelines now. Any other question? All right. Thank you very much for your time. Are there any other textbook references that are available online? There are a number of books that you can get online. The problem is some of them might require subscriptions and things like that. So I will not be able to list for you which ones are available online and which ones are not. However, it then becomes your responsibility to see if when you're looking at the reference list, you can Google to see which one you can get as an online reference. But the problem is with these textbooks, like I think the MEC manual, for example, you can get it online. The only problem is it would require a subscription. So it would then be your responsibility to pay for that if you want to go down that path. As far as the practice papers are concerned, currently we only have those ones which are one per subject available. We haven't been able to make more questions available, and the reason for that is we do not really set the questions ourselves. We outsource, and the outsourcing can become quite an expensive exercise, because then we need to outsource the examiner and then the moderator. And already the way that these exams are expensive, it would become even more expensive, because then we have to find a way to get funding for that exercise. We have been in discussions to see how we can get maybe previous papers available, but we haven't reached the point where we have enough questions for that to be that simple. Are there any tutors affiliated with the SAPC? Unfortunately not. All of our assessors, moderators, examiners, all of them are on contract basis, and we outsource everything. We don't have anyone affiliated with us, unfortunately. Any other question? All right. Thank you again for your availability and your time. This presentation will be made available hopefully next week. Currently, there is the last year one presentation. If you have any question, foreign qualified at sapc.za.org, and we will make sure that we get you the responses that you require, whether it be the question to the examiner or moderator, or a question to the office. Again, thank you very much for your time, and good luck with the examinations. We wish you all the best. We can't wait to have you as interns and serving the profession. Enjoy the rest of your day. Bye.